Good morning. Good morning. Good morning. How are you today? Even better than yesterday, right? Yes. So we start the second day of our workshop, Nestle Nutrition Institute Workshop 94 on milk. On milk, mucosal immunity, and microbiome, impact on the neonate. And I have a great pleasure to introduce you Professor Alan Walker, who is the chairing session today. And session is absolutely fantastic because we will be talking about microbiology of milk and lactation, influence on gut colonization. And Professor Alan Walker, do I need to introduce Professor Alan Walker? Yes. So he is a Conrad Taft Professor of Nutrition and emeritus. And what yesterday Perret said us, this is a unique workshop where we really have a concentration of so high level of expertise, which brings even more and more credibility to the Nestle Nutrition Institute. He's a professor of pediatrics in Harvard Medical School. He has multiple awards, and I believe that he only listed the most important ones. It's Schwachman Award of NASBGAN. It's Award for Excellence in Pediatric Gastroenterology of American Academy of Pediatrics. He is mentor of Research Scholar Award, American Gastroenterology Association Institute, and I may be wrong, Macy Georgi Award on Outstanding Research in Breast Milk Biology. And current interest of Alan Walker in research is impact of breast milk on host defense and initial colonization of the intestinal host defense. With this, I give the stage to Alan Walker, and we are looking forward for a fantastic session. Thank you.
Thank you very much for that kind introduction. It's my pleasure today to introduce our first speaker. I should comment that because this is being recorded, we have to adhere to the hour per speaker. We may have extra time at the end of the day, but this will allow us to get our recordings in. It's my sincere pleasure to introduce to you Samuel Ritava. I have a personal reason for introducing him because he spent several years in our laboratory before he returned to Turku, Finland, where he is an associate professor of neonatology. He's going to give an overview on the milk microbiome and neonatal colonization, and he's had a great deal of experience in this area. Samuel.
Good morning. Thank you very much for the nice introduction, Dr. Walker. It's an honor and a pleasure to be here. Thank you. I would also like to thank the organizers. And it is my task to give an overview of the milk microbiome and neonatal colonization and the current state of knowledge as I see it. What I will be talking about, since I'm a physician, I will begin with health, breastfeeding and health, infant gut microbiota, breastfeeding and health, and those are topics we have already covered, but I can't help but mention those as an introduction to the human milk microbiome, what we know about its composition and origin. We will have separate talks about those, but I will touch upon that. And the last part of my talk is what we know about the significance of the human milk microbiome to infant gut colonization. And I can reveal at this point that we actually know precious little. And what we know about the clinical significance of the milk microbiome, we know even less, and perhaps some ideas for future research. There's only 4 buttons. Okay. Well, I know I'm preaching to the choir here, telling you that breastfeeding is probably one of the most important interventions to improve infant health. There was a beautiful paper in The Lancet a couple of years ago, which I recommend to everybody who hasn't seen it. Dr. Verhasselt alluded to this already yesterday. And based on the paper and based on the analysis in the paper, it's been estimated that the risk of death of an exclusively breastfed infant is only 12% of that of a formula-fed infant in the developing world. It's been estimated that breastfeeding could prevent about half of all diarrheal disease and about 1/3 of all respiratory infections. And consequently, exclusive breastfeeding, could save more than 800,000 lives annually. So it's really the perfect intervention we have at our disposal. And these results are actually kind of easy to understand. We heard beautiful talks yesterday about the protective molecules in human milk. So protection from infections during breastfeeding is probably largely mediated by those protective factors. I think it's also safe to say that in many parts of the world, an infant who is exclusive breastfed is not exposed to contaminated water to the extent that the formula-fed infant might be. So that might be a simple mediating factor as well. However, what is perhaps more interesting and more baffling is the long-term health impact of breastfeeding. It's been estimated that the risk of overweight and obesity is about 1/5 smaller in infants who have been breastfed in later life. It's been estimated that breastfeeding could prevent about 1/3 of all cases of type 2 diabetes, which becomes manifest at the age we start to gain weight and become middle-aged, so much later than breastfeeding. There's no data on supporting the notion that breastfeeding might actually have an impact of other metabolic manifestations such as hypertension or dyslipidemia. It's interesting that breastfeeding has been linked to reduced risk of leukemia, reduced risk of type 1 diabetes. The jury is still out in terms of asthma and atopic dermatitis and allergies. We heard beautiful talks yesterday. There's some suggestion about breastfeeding having an association with the risk of autistic spectrum disease and inflammatory bowel disease. But these are very, very difficult things to study. The diseases become manifest much longer, so we need long follow-up times. And as we all know, it's both practically and ethically impossible to do randomized trials with breastfeeding. So we have to rely on observational data, which is confounded after confounding. In terms of obesity, we know that the most important risk factor for childhood obesity is having an obese mom. We know that obese moms tend to breastfeed less, and shorter periods of time, they tend to give birth by elective C-section. They pass on their microbiota, they pass on their dietary habits, so it's really difficult to tease out what the impact of breastfeeding might be in the whole context. We heard yesterday that breast milk contains live cells and immunomodulatory molecules that might program or modulate immune maturation in the infant, and that's a hot area of research, but it's very, very difficult to tease out what molecules are important and which molecules play a role in terms of any given disease. It's also important to note that breastfeeding is not a controlled substance in the sense that different moms have different type of breast milk, and we heard that breast milk is different at different stages of lactation, but we also know that different conditions, maternal characteristics have an impact on the content of breast milk. We know that allergic moms have different immune molecule concentrations in their breast milk than non-allergic moms. So it's pretty much a mess. So that's why I'm concentrating on a simple thing, the microbiota, which is actually by no means simple. We know, and what I find really interesting, that the gut microbiome has been related to the development of a lot of chronic diseases later in life. There's a list that kind of resembles the diseases that have been linked to breastfeeding. There's atopic disease, obesity and its comorbidities, necrotizing enterocolitis, which is important for me as a neonatologist, but also chronic inflammatory disease like inflammatory bowel disease, type 1 diabetes, some neuropsychiatric disorders. Now, showing that the gut microbiota in a person with a disease or disorder is different from a gut microbiota in a person who doesn't have the disorder is a long way from showing any causal relationship between the two. And what I find more interesting is the studies showing that very early life gut microbiota is already different in individuals who later develop disorders and diseases like obesity, overweight, atopic disease. And we're getting more and more studies like that, and we need studies like this, and they need to be combined with more mechanistic reductionist animal and laboratory models. But anyway, there seems to be a connection with the gut microbiota and chronic disease, and there seems to be a connection with breastfeeding and chronic disease. So we have two connections. Maybe they are the same connection to some point. Well, as we all know, breastfeeding has an impact on the gut microbiota. There's a graph from our Swedish friends from a few years back showing the gut microbiota development in infants during the first 24 months of life on the very general phylum level, and we see these huge changes taking place in gut microbiota composition, especially during the first months of life, which coincide with breastfeeding, the introduction of solid foods, and withdrawal of breastfeeding. There was a beautiful study in Nature last year from the TEDdy study showing that actually exposure to breast milk is the most important determinant of gut microbiota composition in infancy. And interestingly, it's actually the cessation of breastfeeding that causes significant changes, more than the introduction of solid foods. So there's something in breast milk that actually has an impact on gut microbiota. There are many papers showing differences in formula-fed and breast milk-fed infants, and we know that the gut microbiota of breastfed infants seems to be dominated by bifidobacteria. This has been the conventional wisdom for a long time, and it still seems to hold true. Interestingly, one of the members of the audience has shown a couple of years back that actually the impact of breast milk on gut microbiota is dose-dependent. And we know a great deal about the factors that promote the growth of bifidobacteria and certain other bacteria in the infant gut. We will have talks on human milk oligosaccharides, but there are also protein substances in breast milk that promote the growth of bifidobacteria. So what is the role of human milk microbes in this context? As I warned you, we don't know a great deal, but we know something, and that's what we're going into. So the human milk microbiome, this is something we have been talking about for about a decade and a half. To this date, about 200 bacterial species have been detected in milk. And this is just from a study we published this year. This is about, I believe, 40 moms. And if we look at the composition of breast milk microbiota in this study, we see the major phyla, Actinobacteria, Bacteroidetes, Firmicutes, and Proteobacteria present. And on species level, there's a great deal of Staphylococcus, Streptococcus. And there's also a great deal of, as of yet, unidentified bacteria. If you look at different studies, the human milk microbiome composition seems to vary. We don't know if it's because of the stage of lactation or the geographical location. There are a number of things that have an impact on on the milk microbiome. But also we need to think what we are actually measuring when we're measuring the microbiome in milk. The microbes in our milk samples may be derived from the mammary epithelium. There's good data showing that even the non-lactating mammary epithelium harbors a microbiome, and people who are interested in breast cancer are actually interested in the role of the the mammary gland microbiome in the development of cancer. But when we take samples, we also get bacteria from the skin, and actually we might get bacteria that originate in the infant mouth because the child sucks on the nipple, and we collect the milk that comes from the nipple, so we might actually be looking at infant mouth microbiota. Interestingly, however, in addition to Streptococcus and Staphylococcus, we constantly find obligate anaerobes like bifidobacteria, Bacteroides, Clostridia in milk, which in my mind is some indication that we're actually measuring a microbiome specific to the mammary gland and not maybe so much the areolar skin. So we know something about microbes, factors that might have an impact on the milk microbiome. This is a study by Maria Carmen Collado and co-workers from a few years back, and they looked at the milk microbiome and they found interesting things. First, if we look at, they collected samples from a large number of sites in the mother. They collected maternal fecal samples, gut mucosal microbiome samples, skin samples, oral samples, vaginal samples, and milk samples. And in a simple principal coordinate analysis, they showed that the milk microbiome was very distinct from maternal feces, maternal skin, or maternal mouth, and even the birth canal. So there's a distinct microbiome. And interestingly, they had some moms who had delivered by elective C-section, and those milk samples were clearly distinct from the vaginally delivered mom's milk samples, and they resembled perhaps the skin. And when they looked at on the family and genus levels, it's a little bit small text, but here we have on the family level, vaginally delivered, mothers who delivered vaginally, mothers who delivered with C-section, and elective C-section. And the elective C-section milk microbiome was distinct on the family and genus level, whereas the vaginally delivered, mothers who delivered vaginally or the mothers who delivered by C-section after the onset of labor, there was really no difference. So this is an indication that something might actually be going on during labor that has an impact on the milk microbiome, or that labor is needed for a normal milk microbiome, and we'll get back to that. The other interesting observation in this study was that obese moms seem to have a less diverse milk microbiome. So the maternal metabolic state seems to have an impact on the milk microbiome. We do not know what the significance of that is in the development of obesity in the child. Other things that might have an impact on the milk microbiome, this is a small study we did with Dr. Bode, who's here in the audience. We measured from a small number of mothers. We measured the milk microbiome just by qPCR, certain selected species, and we measured human milk oligosaccharide concentrations. And it looks like human milk oligosaccharide concentration correlates with the bacteria in milk. And interestingly, things like Bifidobacterium longum were positively correlated with LNT, which is an oligosaccharide we know that specific bacterium is capable of utilizing. So it's tempting to think that already the milk microbiome is shaped by HMOs. We have really no data to show what the direction of causality is, but it shows that this is a complex system, and already in milk we see these correlations with microbiota and oligosaccharides. We need to continue that line of investigation. Other things, when we think of things that might have an impact on maternal microbiota and the maternal milk microbiota, is of course antibiotics. We saw the studies that the mode of delivery has an impact on the milk microbiome. We started thinking maybe it's not the mode of delivery, maybe it's actually the antibiotic prophylaxis often given in C-section. So we did a study. This was about 40, 50 moms who had delivered vaginally with or without antibiotic prophylaxis for group B strep, who had delivered by C-section or elective C-section with or without antibiotics. At that time in our unit, there was no universal antibiotic prophylaxis for elective C-sections. Now there is, but this was at a time where we're able to actually tease out the impact of antibiotics and C-section. And here we see in a principal coordinate analysis that the light blue and red dots really cluster together, and they are the vaginal delivery ones with or without antibiotics. So we don't see a whole lot of effect of the antibiotics, but the C-section babies cluster differently. So it's not so much the antibiotics, it's more the mode of delivery. When we looked in more detail, we see that the intrapartum antibiotic does have some effect, but these are milk samples that were collected at 1 month of age. So we see only traces of the antibiotic effect. The richness and diversity in the antibiotic-exposed moms is a little bit higher. So that's, again, one of the things that might have an impact. And intrapartum antibiotics is one of my favorite subjects because we know that we need intrapartum antibiotic prophylaxis to prevent group E strep disease. There's no question about that, even though the clinical studies are not so great. But after the adoption of screening practices and prophylaxis, we have seen a massive decline in group E strep sepsis. What I'm more worried about is the prophylaxis for C-section and for other reasons. And if you look at the Cochrane review on the impact on the efficacy of intrapartum antibiotic prophylaxis or prior to C-section, they said that it prevents wound infections. It prevents uterine infections in the moms. And there's a horrible sentence that says the impact on the child is unknown. And for somebody who's taking care of the child, I think that's a horrible sentence. So we are in the process of looking at what intrapartum antibiotic might do to the child. And we are looking at the larger cohort. I don't have that data to share yet, but we did a small study of babies with infantile colic and compared them to healthy, non-crying infants, and we could see that all of the prenatal and perinatal factors that might be related to colic, we saw that intrapartum antibiotics increased the risk of infantile colic by a factor of 5. And the only protective factor we saw was exclusive breastfeeding. Again, it's intrapartum antibiotics, it's exclusive breastfed. We know that colic is somehow probably related to the gut microbiome. So again, more questions than answers, but I think this is a very important area of study. Okay, moving on. So we know that there is a microbiome in milk, and we know that it is different, related or associated to certain maternal factors, obesity, mode of birth, antibiotics, HMOs. What about, where does it come from? And I know there's a separate talk about this, so I'm not going into detail, but we have some fascinating ideas. As I mentioned earlier, we know that we find obligate anaerobic bacteria such as Bifidobacterium, Bacteroides, Parabacteroides, and Clostridia in human milk. And those are usually associated with the gut. So it's a tempting idea that there might be an entry memory pathway for microbes as well. We know from clinical studies where we have studies in which probiotic bacteria are administered to the mom to prevent disease in the child or to prevent or treat mastitis. Investigators in Sweden and in Spain have been able to find those specific probiotic bacteria in milk samples. It might be contamination from the skin, but I think it's tempting to speculate that they actually go through an intramammary pathway, end up in the milk. We have done quite a few studies on pregnant and breastfeeding women using probiotics, Lactobacillus rhamnosus GG, certain bifidobacteria, and I have to say we have tried hard, but we have not found those in milk. I would have loved to find those in milk, but we can't. But other investigators who are probably better than us have found them in milk, and I think that is really fascinating. If we think of the intramammary pathway, there are nice studies showing that in experimental animals, in the perinatal period, the gut becomes more permeable. And you might remember I showed you data that being born by elective C-section without the onset of labor has an impact on milk microbiota composition. I think it's intriguing to speculate maybe that happens in humans too, and if you don't go through labor, your gut doesn't open up and the intramammary pathway might not be functioning. This is all speculation, but I think it's really interesting speculation. This is an old study from 12 years ago, and investigators in France looked at maternal fecal samples in a small number of moms, 4 moms, maternal fecal samples, blood samples, milk samples, and infant feces samples. And with the methods they had available at that time, they were able to find certain
strains of bacteria here, two Staph epidermidis and Streptococcus salivarius. They found them constantly in maternal feces, in maternal blood samples, in milk samples, and in the infant gut microbiome. Again, and B. longum. And again, this is no direct proof that that is the way the child acquires those bacteria, but I think it's interesting that they were actually able to stain those bacteria in maternal cells in human milk and in lymphocytes in maternal, or in immune cells in maternal blood. So I think this is a really fascinating area of investigation. Okay, I'm not going to talk more about the origin of the milk microbes since we have a separate talk on that subject. So now let's move on to what I was actually supposed to be talking about, the impact of the milk microbiome on infant gut colonization. And I've been stalling because there isn't a whole lot to say, but there are some nice studies. Whether we can prove that the microbes in milk actually colonize the infant or not is a matter of, we need really sophisticated studies. Thus far, we have really only done studies where we collect samples from the mom, from maternal gut, from milk, and from the infant mouth and gut, and see correlations. And this is pretty much where we are at. This is one of the first studies that this was conducted in Finland already more than 10 years ago. This is about 60 moms and maternal fecal samples, human milk samples, and infant fecal samples were collected, and the presence and abundance of bifidobacteria were looked at by qPCR at that time, and constantly different species or groups of bifidobacteria were detected in all the sample types. So this might be an indication, okay, we find the bug in the maternal gut and in milk and the infant gut. This might be an indication that something is going on, but when they actually looked in detail, there was a strong correlation between the maternal gut bifidobacteria and infant gut bacteria, but there was really no correlation with the milk, with the abundance and presence in milk. So something is going on, but whether the bacteria in milk are just an innocent bystander and not actually a colonizing inoculum, it's hard to say based on this study. Moving on, this is a beautiful study from about 5 years ago. Only 7 mothers, but multiple techniques, a nice sample collection. Again, samples from maternal gut, milk, and infant gut, and 16S sequencing, so more advanced methods, and the data on the phylum level and also on the genus level. And the investigators find the same bacteria present in maternal feces, in human milk, and in infant feces. Interesting, there are some genera like Pseudomonas and Rastonia that are only present in milk, and they are not detected in the infant or in the maternal gut. But what the investigators did, they also cultured, and they, especially Bifidobacterium breve, they found viable B. breve in the maternal gut, in milk, and in the infant gut. Okay, again, circumstantial evidence, something might be going on. The author of this paper is in the audience, so I'm not going to spend too much time. A beautiful study, more than 100 mother-infant pairs, and they looked at the contribution of milk microbiome and the breast skin microbiome, and they could show that in a dose-dependent manner, it seems that they actually, through the use of a source tracker technique, actually in infants who are more than 75% breastfed, about 15% of the infant gut microbes originate from the milk. And what they did is really interesting. At the age of about 6 weeks, the gut microbiome of an infant resembled more closely to his or her own mom's milk than other milk samples, which is an indication that maybe the milk is actually a source of bacteria to the child. Moving on, this is probably the latest study from this year. It's less than 20 kids, but nice sample collection. They collected infant feces samples and from the mom both fecal samples and milk samples, and mouth microbiome samples from the child as well. And if we look in detail, the infant feces here red, it's kind of all over the place as it tends to be, whereas the maternal feces is much more clustered. And we see that the milk microbiome is clearly distinct from that of the maternal microbiota, gut microbiota, and also from the infant gut microbiota. It's actually a bit closer to the oral microbiota, both in the infant and the child. And by source tracker, they showed that on day 2 of life, so very, very early, only about 5% of the gut microbiota originates in milk as per source tracker analysis. And at 6 months of age, the contribution of the milk microbiota to the gut microbiota is really negligible. It's 0.3%. So maybe it contributes more to the oral microbiota, which is an understudied subject. We published a study on the oral microbiota a couple of years back, last year actually. And we looked at 35 moms and children. We looked at the oral microbiota in the child and the breast milk microbiota. And we actually saw that they were quite similar in taxonomic analysis. But of the 22 genera detected in the study, only 8 were shared by both milk and the infant mouth, and the microbes shared were again Streptococcus and Staphylococcus. And when we tried to see whether a child's mouth microbiome was more closely related to his or her own mom than other moms, who weren't able to do that. So it doesn't look like the milk microbiome even contributes greatly to infant mouth colonization. So what goes on very early in life? You might remember from the previous slide that, that we showed that it's more important in the first days of life. This is a study we published a few years ago about 15 mother-infant pairs, and these were all children born by elective C-section. So we wanted to have a uniform group, and we actually wanted to avoid contamination during delivery. And we looked at for the purposes of this talk, we looked at the maternal fecal microbiome, which were clustered nicely in a group and had very little resemblance with the other sample types. We looked at meconium in light blue, and we looked at infant feces at about 1 week of age in dark blue, and we see a shift during the first week of life. If you look close, you might imagine that the light blue dots shift in this direction and perhaps resemble colostrum more closely. So maybe something is happening during the first week of life. What is also important is colostrum. The colostrum microbiome is nothing like the maternal gut microbiome. And if we believe in an intramammary system, it seems to be a very selective system, and this we see time and time again. What about later in the neonatal period? This is unpublished data from Henni Hiltunen, who's a PhD student in Turku. We looked at about 30 preterm infants between the gestational ages of 25 and 35 weeks, so it's a very heterogeneous group of preterm infants, and we did serial samples of the infant gut microbiome. We took samples at 3 days of age, green, which are really all over the place. At 7 days of age, it's a prune-colored dots, and they start to cluster a bit more nicely. And 28 days of age, at 1 month of age, and this is when all babies were receiving breast milk. All of them were receiving some breast milk at 3 days of age, but at 28 days of age, all but one were exclusively enterally fed. And we see that the 28-day fecal samples start to cluster. So the chaotic initial colonization kind of draws toward a cluster at 28 days of age. The mom's fecal samples collected right after delivery on day 1 or 2, the first stool the mom passed after delivery, they cluster nicely, but they don't really resemble the infant gut microbiome. And then breast milk collected at 1 month of age, so at the same time as these yellow samples in red, and it's pretty much all over the place. If anything, it resembles the very early meconium microbiome, but we see really no clear connection with the preterm infant microbiome. Now, these are all infants in the NICU, so it's a very unnatural situation. I think all but 1 or 2 of the infants had received antibiotics at some point. But what we can draw from that is, again, the milk microbiome is nothing like the maternal gut microbiome, and it bears little resemblance to the developing infant gut microbiome. We are doing source tracker analysis as we speak. I don't have that data quite yet. Okay, so we see that something is going on. There's definitely live bacteria in milk, which the baby ingests. They might have some impact on the mouth microbiome. They might have some impact on the gut microbiome. Depending on the study, it's negligible to 15%. And we don't really know why they are there. I've toyed with an idea close to what Dr. Verhasselt told us yesterday, that maybe the bacteria in milk are not there to colonize. Maybe they are there to tolerize in the context of the tolerogenic milieu of the milk. We don't know. This is all speculation. Somebody said yesterday that nothing happens for no apparent reason. I think the microbes in milk are there for a reason. I think we have reason to believe that there is an active system by which they are presented in milk, and they must play some biological role, but we don't really have any grasp of what that might be. Now, if we think of the clinical significance of the milk microbiome, that also remains an open question. I think one of the first indications that the microbes in milk might actually have some clinical relevance is a paper we published last year, where we looked at, this was about 20 mother-infant pairs, and we looked at, again, the gut microbiota in the mom during last trimester of pregnancy and 1 month after delivery. We looked at the infant gut microbiome at 1 month of age and 6 months of age, and we looked at the milk microbiome in colostrum, the first days of life, and 1 month of age. And just by first glance, we see that and this is on genus level, we see that the milk microbiome is nothing like the infant gut microbiome. It's nothing like the maternal gut microbiome, which the maternal gut microbiome seems to be kind of stable in the last trimester of pregnancy and after delivery. The infant gut microbiome undergoes some changes, I would say, from 1 month to 6 months, but the milk microbiome resembles them not at all. And again, we see a lot of streptococcus, we see a lot of staphylococcus, some change over the course of lactation. But what is really interesting, when we looked at the number of antibiotic resistance genes in these samples, we see that infants actually harbor a large abundance of antibiotic resistance genes. And these are term healthy infants who have never been exposed to antibiotics. They actually have more antibiotic resistance genes in their gut microbiota than their moms. And halfway is the milk microbiome. There's a large abundance of antibiotic resistance genes in the mom. And when we put them on, again, in a two-dimensional space, we see that the maternal fecal samples, the antibiotic resistance genes in the maternal fecal samples are pretty stable during the last trimester of pregnancy and 1 month after delivery. And we see that the antibiotic resistance genes in the infants are clearly distinct, and these are not antibiotic-exposed infants, and they really overlap with a profile in milk. So I know that it's not a nice thing to say that milk might be doing some harm, but we actually might be passing our children antibiotic resistance genes in milk. Again, non-antibiotic exposed infants get the antibiotic resistance genes, at least partially, from milk, so they're paying the price for the past antibiotic use in the moms. What is a relief, though, is when we looked at the impact of breastfeeding on the number of antibiotic resistance genes, we see that almost universally the infants who were breastfed at 6 months had less antibiotic resistance genes in their feces. So first I was saying that, okay, they get it from milk, but if they are breastfed, they have less. How does that work? The explanation we have for that is that most of those antibiotic resistance genes are in proteobacteria. They are actually in enterobacteria. E. coli is the most important carrier of antibiotic resistance genes in the infant gut, and the number of those bacteria is lower in breastfed infants. The breastfed infants, again in this study, harbored more bifidobacteria, which were protective of the antibiotic resistance genes. And again, my favorite subject, intrapartum antibiotics, increase the amount of antibiotic resistance genes. So from a practical point of view, we cannot help the past antibiotic use of the moms, and we cannot help how many antibiotic resistance genes the mom carries, and she might be passing them to her infant partially through breast milk. But the two wise things we can do to reduce that is not to use antibiotics when they are not needed and promote breastfeeding. Okay. Luckily, I can turn this into promotion of breastfeeding, but it was a little bit alarming at first. Breastfeed, promote bifidobacteria, decrease the abundance of E. coli and other enterobacteria in the infant gut, and you reduce the amount of antibiotic resistance genes. Well, I said this is the first indication of clinical relevance. Whether this actually has clinical relevance, we don't know yet, but it is alarming. So a lot of things are going on in terms of the milk. Okay. So what have we discussed? What have I tried to convince you of today? Well, first of all, there is such a thing as a human milk microbiome, and it seems to be affected by a number of maternal factors. The maternal metabolic state, maternal overweight, mode of delivery, especially if a C-section is performed without the onset of labor. It looks like the mammary gland doesn't know if the baby is born by C-section or not, but the mammary gland seems to know whether the mother has experienced labor or not. Antibiotics seem to have an effect. What we don't know, in our preemie study, we had a wide range of premature infants, and we saw no impact of the duration of gestation between 25 and 35 weeks on the milk microbiome, but I think we need to study that more. The impact of gestational age, the impact of lactational stage, the impact of geographical area, those are all open areas of investigation. We'll have a talk about the origin of the microbes in milk, but I think there are good grounds to hypothesize that there is an active system by which maternal gut bacteria are transported into milk. Why that may be, we have little or no understanding of. How the milk microbes affect gut colonization is an open question. There's some indication they might be. There's some studies showing that they really have no impact. My pet hypothesis that they are at least partially there to tolerize the child to the microbiota. The child is going to have absolutely no data to back that up. At this point, it's a hypothesis. And the whole clinical significance of the milk microbiome is an open question, and we can say that it's frustrating we know nothing about it, but I think it's motivating that we know nothing about it, and we should really get to work and find out why the bacteria in milk are there. In the end, I'd like to thank all the people who actually did the work from our lab, which I showed. I borrowed a lot of stuff from a lot of authors, but these are the people who directly contributed to the papers from Turku. Thank you very much.
This is open for discussion, right?
Very interesting and very important studies. Thank you for sharing it with us. I have two important issues to consider. One is that is the microbiome cell-free or cell-associated? Because there is such a large quantity of particularly cells, epithelial cells, macrophages, and others. Do you think what you see is really cell-free material, or have you made an attempt to see whether there's a lot of cell-associated organism there? That's one question. And the second question I have is, there's a lot of antibody activity in the milk to many of the organs that you talked about. Have you looked at association of immune complexes and tried to break it down in terms of how much of organisms are antibody-free or antibody-associated in the milk?
Thank you. Those are excellent questions, and I think interrelated. We haven't done any systematic studies in that fashion. I think we need to do that. I think at this point, when people are relying on sequencing methods, they just isolate DNA, and we have ways of trying to see whether there are intact bacteria or bacterial DNA, but we should also be looking at whether the bacteria might be intracellular, as you say. And there was the old study from France showing that they actually can stain the bacteria in cells. And the next question would be that if they are free-floating, are they coated with antibodies? And I don't think, at least I am not aware of any study where that has been looked at, but that would have a huge impact on their effects and immunostimulation in the gut.
Jerry?
I'm actually continuing in the question which you made. We live with millions of different, or trillions, quadrillions of different bacteria, but they are not free. All bacteria in our oral cavity in the gut are coated in vivo with antibodies, which is a factor which you never consider in any of the studies. And yet, as I showed yesterday, the interaction is mediated by specific antibody activity or carbohydrate interactions. Now, you showed us cells which apparently have bacteria in it. What cell type is it? Is it phagocytic cell? They express Fc alpha receptors. Those bacteria are swimming in oceans full of IgA. Have you ever considered that may have tremendous importance not only for internalization of bacteria in milk in phagocytic cells, but also in the child? Because bacteria coated with IgA certainly behave differently than a free bacterium in the gut. That would penetrate, coated with IgA will not. So are any of you looking at the effect of antibodies in the colonization and protection, for heaven's sake?
Thank you. Your point is extremely well taken. I don't think anybody has systemically done that. We're in the very first stages, but that's a very important point we need to consider.
That will explain so many things.
Yes.
Thank you very much for your nice presentation. Today we are breathing a bit easily, being as pediatrician. Yesterday it was all talks were, and in research level and molecular level, but today we are feeling a bit relaxed. And my first question is that you have told that giving a mother to Lactobacillus reuteri and cerevisiae to the mother. So is there any difference if you rub this bacteria, probiotics, to the areola and the baby suck it directly, or if you give it to the mother and then coming all the traveling to the mammary cells? Is there any difference of the bacteria behavior if you give it directly to the areola or to the mother?
Well, this is really beyond my expertise, but I think if the mom ingested that, it's been processed by the mom's immune system, and it might be delivered in an immune cell, in a phagocytic cell, in an APC, and it might be coated with antibodies. And if you rub it on the areola, I'm not sure how well some of those probiotic bacteria survive, but I know there are studies improving child oral health where they have actually a pacifier, which releases the probiotic directly in the mouth. So I think the areola might be rubbing the areola, which I do not recommend, might be a similar approach.
I have a second question. As you have mentioned about premature babies, in many cases of premature babies, the mothers are sick. Maybe it's eclampsia, and or multiple pregnancies with multiple premature babies, and they are really, devoid of breast milk. Is not available. So sometimes we use a surrogate mother, but though the composition of the milk is not appropriate for the premature baby if it is collected from the surrogate mother. In that cases, milk fortifier can be used for premature babies. But I do not know, the Nestle, this question may go to the Nestle too, they have preparations for premature formulas, but they are not coming up with the milk fortifier, so that we can use the milk fortifier and the milk from the surrogate mother to provide for the premature babies whose mother is really sick or eclamptic. So Nestle can answer this question, or you?
Well, that's a number of excellent questions. In our small preemie studies, we looked at a number of maternal factors and their impact on the milk microbiome, and we see nothing, depending on the cause of prematurity and a number of things, mode of birth, we see absolutely nothing in the premature baby microbiota. I'm not sure if the premature baby needs the microbiome, but I'm not familiar with using surrogate moms.
Milks. Fortifiers. Fortifier is not the microbes. These are micronutrients.
Right. Yeah. Right.
Yeah.
Well, there's a point I want to make about using a surrogate mom, and you said if the surrogate mom has not given birth to a premature baby, that might not be appropriate milk from a premature baby. There is a notion that premature baby moms of premature babies have different milk in composition, not only in microbiota, but in other things like EGF, and that's an adaptation to serve the premature baby. I have a very hard time believing that. In order for that to be an adaptation, it would have had to offer a survival benefit. And before the last century, I don't think any less than 32-weeker survived in order to actually have that evolutionary drive. I don't think the premature babies survived. There can be an adaptation in human milk. I think it's more likely that it's a sign of an immature mammary gland at that stage of pregnancy. So I think, in my opinion, it doesn't matter if you use a donor milk or a surrogate mom, whether the donor delivered a premature baby. I think we should pay attention whether we give colostrum or early milk. We don't have a lot of donated colostrum, but we should pay attention at which stage of lactation the milk sample has been taken. Now, in terms of fortification, there are fortifiers you can use in human milk, and it doesn't matter if it's a, I'm not sure if I understand your question, because you can use them on a
It's not available everywhere. Milk fortifier. It's not available in our country.
Okay.
But the premature formulas are available, Nestle's are.
Right, okay.
Yeah.
I didn't appreciate that, and I'm a simple neonatologist. I'm sorry I can't help you, but that is a problem.
Carlos?
Yeah, in continuation with the idea of the probiotic not appearing in breast milk, I wonder if anybody has done like the labeling of the probiotic with nitrogen 15, for example, which is a non-radioactive isotope, and then seeing enrichment in breast milk just to see if the protein of the probiotic, might not go beyond past the lymphatic system, but it could contribute in some way to protein or something in breast milk.
That's an excellent question, which I can answer, and I direct it to Dr. Rodriguez, who will give the next talk. Maybe you can address it.
Maybe better after the next talk. Yeah. Okay. Ferdinand?
I would just like to clarify the availability of human milk fortifiers. Human milk fortifiers are globally available from 4 companies at least. They are available in liquid form and in powdered form. If they are not available in your country, it's an effect of year-long fighting with the regulatory authorities in the respective countries which do not accept the fortifiers. It is also something which is very difficult in terms of the interactions with NGOs. Some NGOs in the countries, mainly in South Asia, they believe that this interferes with breastfeeding. To a certain extent, they are right, because the fortifiers are difficult to handle. The mother has to pump the milk and then mix it and give it in a bottle. So this is not something where companies are not willing to distribute fortifiers to the countries. It's mainly the interaction with the regulatory authorities which do not allow to have them in your country.
At the same time, they are giving permission for the premature formulas.
The formula is a different thing. A formula is a different category of special food for premature infants. One is a supplement and one is a whole nutrition. So the authorities see it in a different way.
Are there any more questions? If not, Sam, thank you very much.
One question. One question.
One question, please. You know, that light is shining right in our eyes.
Yeah.
I can't see that. Go ahead.
Thank you for this very beautiful synthesis. I had one comment and one question. The comment was regarding the IgA and microbiota. So there are papers showing that the IgA, of course, shape the microbiota, but also influence the metabolism of the bacteria. So it really goes far. And then a question we had yesterday talks about evolution of breast milk and how is it spread among species. Have you ideas about or knowledge about microbiota in breast milk of other species? Is that also found? And
I have to confess I have absolutely no knowledge of that.
Are there any other questions?
Yeah.
Okay, go ahead.
In case of necrotizing enterocolitis, actually we don't give the best feed for certain period of time. On that time, could be probiotics be given for the prevention of the necrotic enterocolitis?
Now that's a discussion no neonatologist wants to enter. Yes. Well, I believe there are very few instances when a premature baby shouldn't receive breast milk. And those are instances when we are afraid to use the gut at all. And I think at those times, I wouldn't give probiotics either. When the baby receives breast milk, I think if you are among those who believe in probiotics, whenever the baby is receiving breast milk, you can give probiotics as well. But if the baby is NPO for a reason related to the intestine, I wouldn't give probiotics. But that's my opinion. I'm not even sure if that's an expert opinion, but that's my opinion. I'm not sure if Dr. New has other insight.
I'll talk about it later.
Thank you very much, Sam.
Thank you.
We're off to a very lively discussion. It's my pleasure to introduce our next speaker, Professor Juan Rodriguez, who is professor in the Department of Nutrition and Food Science at Complutense University in Madrid. Now, he may have a lot of claims to fame, but the one that I think is most important, he was the first person to describe the breast milk microbiome. And he's going to talk to us today about human milk microbiota origin and potential uses. Dr. Rodriguez. Oh, I'm sorry. I beg your pardon.
Otherwise, people will fall asleep.
I'm trying to shorten this up. Go ahead. Have a coffee break first. Good morning. It's my pleasure to reintroduce Dr. Juan Rodriguez, who is a professor of Department of Nutrition and Food Science at Complutense University in Madrid. And as I mentioned before, he's got the unique honor of being the first to describe the microbiota in breast milk. He's going to talk to us about human microbiota and some of their potential uses. Juan.
So, well, first of all, thank you very much. Thank you very much, Alan. Thank you very much to the chairs, to the Nestlé Nutrition Institute for the kind invitation to participate in this excellent meeting. So it's a good opportunity to learn from people with a lot of expertise in related fields. So I divide my presentation in three main parts. The first one is very short, is a few slides talking about the human milk microbiota, but some aspects that I think they are important to investigate the origin of the human milk microbiota. Then I will talk the main part of the talk about the origin of this bacteria, and then finally, if you are still awake, then I will talk about some uses of the human milk bacteria, and especially for breast health. So this is the year 1988, is the year in which I first started working with human microbiota. And then at that time, it was considered that only a few places in the human body has a microbiota. At that time, we call a microflora, these kind of things. So the oral cavity, the skin, the gut, the vagina. This is a magazine, a very popular magazine in Spain in this year. So they talking about the benefits of human milk, saying that it's a sterile food always available. And it's funny, just talking about the benefits of human milk, they show the photograph of a bottle-feed baby. So this is last year, 30 years later. So you can see the literature. So we can find microbiome almost in all our body, in all our mucosal surfaces that produce a biological fluid and is open to the outside. Of course, there are some microbiota that are controversial, such as that in the lungs, for example, and all others that are very, very controversial, for example, in the placenta and this kind of stuff. So anyway, I think it's important to note that the different ecosystems in the human body are very dynamic and are open to exchange between them. So now it's widely acknowledged that human milk has microbes, and these are among the first microbes that a baby receives after they are born. And in fact, the human milk microbiota is now studied in several labs around the world. And it's also important to note that the data that we have got so far show that this is not a random contamination, but a specific microbiome with a specific structure and with well-defined consortia. So just very quickly, as has been told before, by culture-based methods, we have been able to isolate a lot of genera, a lot of different species, including new species, and other ones will come very, very, very shortly. So mainly, they belong to these genera, Staphylococcus, Streptococcus, Propionibacterium, Corynebacterium, and these bacteria are commonly isolated worldwide, from for most women worldwide. Sometimes we can also isolate lactic acid bacteria and Bifidobacterium, but we have found them to be common in developing countries, for example, where there has been no use of antibiotics previously. Just as an example, for example, samples from Mozambique, samples from the Central African Republic, we have been able to isolate up to 12 different lactobacillus species. And it's something that we had never seen in samples from Spain or from Germany or from Austria, from the States or from Sweden, for example. So this will be a kind of core microbiota of human milk. Of course, when we study DNA, we confirm the presence of DNA from such bacteria, but also, as stated before, we can also detect DNA for gut-related street anaerobes. Of course, they are not cultivable or very difficult to culture at present, so we cannot find by culture methods. So maybe this could be the core microbiome of human milk, but also we detect a third group of bacteria that are usually related to the soil and to water. And this is important because some papers in the past, and including some from our own group, we found that such bacteria can belong to the core microbiome of the human milk. And in the last years, we have realized that maybe they are technical artifacts. Why? Because molecular biology regions have a DNA from such bacteria, and is a problem worldwide. It's difficult even today to use DNA, bacterial DNA-free molecular biology regions. So its presence is very relevant in genomic analysis in low-abundance microbiomes, like that of human milk. So because contaminant that bacterial DNA can amplify even at higher concentration than the DNA of the bacteria that are naturally found in this biological fluid. So this means that our previous studies maybe can lead to wrong conclusions. And this also highlights the importance of the plant controls, also the so-called mock communities. So communities of, we prepare DNA from bacteria that we grow in the lab from different species at a known concentration, and this is our best controls for microbiome studies of human milk. Anyway, when we talk about the human milk microbiome, actually, at the end of the day, we are talking about bacteria. But in human milk, there are other kind of organisms that can be quite relevant, but we don't know. For example, there can be viruses, and the talk after mine, we'll talk about that, archaeas, molds, yeast, protozoa, but of course, most of the studies has been focused only on bacteria. And we found different phyla, for example, Bacteroidetes, Firmicutes, Actinobacteria, Proteobacteria, and Verrucomicrobia. But it's very important to have in account that the highest diversity in human milk is not provided by phylum, at the phylum level, is provided by strains. And this is actually a real example from our lab. In a single milk sample, of course, we have one phyla, one phylum, Firmicutes, one genus, Staphylococcus. We found two species, Epidermidis and Hominis, but we found in the same sample 12 different strains of Staphylococcus epidermidis and 4 different strains of Staphylococcus hominis by pulse field gel electrophoresis and also by multilocus sequence typing. And of course, each strain provides complementary functions. They have redundant functions, but also complementary functions. So the next slide is a kind of silly slide, but just to show the importance of the strain level. So these are human strains, no? Even some of you can be found here, around here. So there are no human beings that are identical. All of us, we have some properties, so things that we do better, things that we do wrong. So of course, at a lower genetic complexity, for example, different strains of Staphylococcus epidermidis has different properties. So this is important, for example, for the origin of bacteria. When we are talking about the origin of bacteria, we have to show a transfer of bacteria at the strain level, not at the species level, the genus level, or the family level. So I'm going to talk now a little bit about the origin of human milk bacteria. As we know, there is a huge potential of mother to infant transfer of bacteria. So through different routes, I told some of them controversial, but other well-defined typical routes. So the child not only inherits the color of the eyes, not only inherits the genes, the human genes. Of course, there is also an inheritance of bacterial genes because they are transferred from mother to baby. But of course, apart from the mother, microbes can come from everywhere we can have in mind. So the rest of the family, sometimes a nasty experience, sometimes good. But of course, any other place from the environment, from other animals, or I don't know, from the air, anything you may have in mind. And of course, breastfeeding is a very close, a very strong, very intense physical contact between the mother and the infant. So anything you want to see. And of course, all the babies, either born by vaginal delivery or born by C-section, they acquire bacteria when they are at that time. And of course, for example, there are previous studies showing that there is a certain degree of milk flow back within the gut after milk ejection. So, for example, when we think about Streptococcus, milk is very rich in Streptococcus, but particularly of two groups, the Mitis and the Salivarius Streptococci. So when we think about these microbes, we always think in infant mouth or in the nasopharynx, but there are not many studies. It's just our belief. We think that this is the logical source of this kind of bacteria. And of course, also some related gram-positive bacteria, such as Rhotia, Cocuria, and so on. However, Streptococci are not only living in the oral cavity. They are living in all the digestive tract. We can find them in the colon, we can find them in the stomach, we can find them at different places. And we got some data in our lab showing that there are Streptococci in colostrum before the first feeding. So we don't know if they come from the infant mouth to the milk to the infant mouth, and most probably there are two ways just at the same time, simultaneously. But anyway, just a few months ago, we published this paper working about the microbiota of human precolostrum. And then we collected samples from 17 mother-infant pairs, samples of precolostrum, so this kind of liquid produced before delivery in a few percentage of women. We also collected oral swabs of the babies, and then we studied by culture-dependent, growing in Columbia agar and identifying the isolated by Malditov, and also a microbiome study. And look, you can find that most of the genera that we found in the infant mouth were already in the precolostrum samples. And when culture-dependent studies and genotyping, we showed a potential sharing of a strain in 14 out of the 17 pairs. Not only Streptococcus salivarius, Streptococcus mitis, but also, for example, Staphylococcus epidermidis somewhere here. So we did whole genome sequencing of all the isolates that we found at the species level, both in the maternal and the infant sample. And we found that in most of the mother-infant pairs, there were streptococcal and staphylococcal strains isolated in precolostrum and later in the oral mouth, including new streptococcal species that will be published shortly. So we know that this way may occur. And of course, this way can also occur, and is logical. But there is no studies showing this way so far, and I hope we can also provide data in the next months. Of course, the second source of bacteria for it has been told, and I told as well, that the human milk is very rich in Staphylococcus, particularly in coagulase-negative staph, mainly Staphylococcus epidermidis, but also in Propionibacterium corynebacterium. And of course, for example, Montgomery glands, there is an hypertrophy, there is during pregnancy, and it contains this kind of bacteria. And you know, the skin in the nipple, in the mammary areola is very raw, so it's logical to think that there is a transfer of this kind of bacteria from the skin to the milk. However, just to think about that, why Staphylococcus epidermidis is always considered just a skin species? And everywhere you find a Staphylococcus epidermidis is a skin contamination. Please just have in mind that the Staphylococcus epidermidis is called this because it was first isolated in a skin sample. But this can be isolated from the mouth, can be isolated from the gut, from the milk, from the vagina, from almost everywhere, from meconium, from amniotic fluid, from the prostate, from the seminal glands. So, of course, and also have in mind that the highest concentration in a human body of coagulase negative staph are in the gut, are not in the skin. Of course, we have in the skin as well. Anyway, independently on where Staphylococcus comes from, we have to know that the mammary environment is quite suitable for this kind of microbes. Just think that the mammary gland during late pregnancy and lactation is a very complex ductal system and is a kind of catheter-like system. And we know that the Staphylococcus staphs are a high problem in the NICUs and other places because they can contaminate the catheters and these kind of things. So from an ecological point of view, are in the best place to be. But also, we know that the mammary gland produce a lactose-rich biological fluid. And Staphylococci and Streptococci, by evolutive genomics, we can see that they are the microbes with the most efficient galactose and lactose metabolism. And also, mammary gland also produce a human milk oligosaccharides. And also, this kind of bacteria, for example, coagulase negative staph, can use quite efficiently human milk oligosaccharides. Of course, so at the end of the day, it's not strange that Staphylococci may be the dominant bacteria in the fecal samples of breastfeed babies for the first weeks of life. 15 years ago, we published also this paper, and just describing the presence of lactic acid bacteria in human milk. And these are bacteria that we didn't expect to find in human milk because we don't find in the skin, we don't find at the beginning in the oral mouth, so the origin was quite puzzling for us. So just knowing that, we, in a collaboration with a hospital, the neonatal unit in a hospital in Madrid, they collected a lot of perinatal samples to try to elucidate where these microbes can come from amniotic fluid to vaginal swab, any kind of material. And when our attention was focused only on lactic acid bacteria, and of course, genotyping at the strain level. And then we found that the profile of this bacteria in vaginal exudate in the breast skin and the milk was completely different. For example, in the vaginal exudate, we found Lactobacillus crispatus, Yerseni, Iners, Gasseri, and so on. In breast milk, in breast skin, we could not isolate lactobacilli, even with prick tests and this kind of techniques. And in the milk, we also found different species and different strains. However, we find a sharing of strains in maternal feces, in milk, and also in the infant feces. And later on, we show, for the first time as well, the isolation not only of lactic acid bacteria, but bifidobacterial strains that are strict anaerobes. So the presence in breast milk is even more puzzling than that of lactobacillus, because are strict anaerobes, this kind of things. So bacterial communities existing in human milk, skin, and oropharynx may share some phallotypes, but also major difference can be observed. So where these differences, where the bacteria can come, our initial data suggests that maybe the answer was in the gut. Nowadays, we know that, for example, the oral mouth or the oral cavity, the mouth, can be also a relevant source of bacteria. But at that time, anyway, we didn't know at this stage how some bacteria may reach the mammary gland. So in the year 1999 and the year 2001, a couple of papers appeared that was a kind of light for us. Particularly this paper showed that dendritic-like cells can sample commensal bacteria in the lamen of healthy host, then go to the lamina propria and spread to distant organs. So immediately, the same year, we sent a student of our lab to the lab where this model was proposed, just to study if this could happen, could explain at least the spread of some bacteria from the gut to the mammary gland. Our idea was that if there was a kind of bacterial enteromammary route, so if some bacteria may interact with dendritic cells or with macrophages, this kind of bacteria, and then go across the mesenteric lymph node and through the thoracic duct, then appear in the milk. Just having also in mind that at that time we were participating in a European project just trying to create a collection of referent lactobacillus strains. And in this collection, there were more than 400 different lactobacillus strains, and almost a quarter of them had been isolated for human blood of healthy people, and they belong to 16 different species. So it seems that the dendritic cells was in the middle or were a key element for our explanation. So in the year 2001 as well, I have a meeting in London with Ralph Steinman. I think maybe some of you maybe know as well. I think maybe he's the only person winning the Nobel Prize that didn't know that he won the Nobel Prize, because he died 3 days before the official communication of the academy. But anyway, we have a meeting with them, 30-minute meeting, and we present our data, and he said, this makes sense to me. I don't know if you are right or you are wrong, but it deserves to be searched. So just go ahead, and then we go ahead. We went ahead. So you know, dendritic cells are among the professional antigen-presenting cells. When they are not non-active, they have a round shape, and they are there for antigen sampling and uptake. But then when upon activation, for example, for the presence of microbes, these dendrites are created, and they get migratory ability. So our first objective was to know what happened when we put together in the same sample a strain isolate from human milk and dendritic-like cells. We have studied this using 4 different dendritic-like cell lines. One from the spleen of mice, one KG1 cell line from the lab, and also in 2 cases human milk, dendritic-like cells obtained from human blood and differentiated in the lab. But the results have been quite similar with all the four different types of cells. So we put together immature dendritic cells and different bacteria isolated from human milk, and we found that just after a few minutes appear to have these dendrites, this kind of thing, these changes in the morphology. But also from the functional point of view, we check to some proteins that are characteristic of activated dendritic cells, such as the major histocompatibility class system of class 2, and also the co-stimulatory B7 protein and other kind of proteins. And we saw that in most of the cases, a large quantity of the dendritic cells became activated. They express these markers. So we saw our results suggested that the contact with this bacteria with immature dendritic cells activate them to mature dendritic cells. So our second objective, just to see if lactic acid bacteria can translocate through an epithelium in the absence of dendritic cells. In this kind of transwell bicompartmental system, we add bacteria in the apical compartment, and then what we saw is that there is no bacterial translocation in the absence of dendritic cells. For example, in contrast, when we add a heteropathogenic E. coli strain, there was a massive translocation, and we can find a high concentration in the basolateral compartment. So the following step was to know if there is a dendritic cell-driven bacterial translocation. So in the same model, we added dendritic cells labeled with orange tracker. We added bacteria labeled with FITC, with a fluorescent, and so just to check if this may happen. We check by flow cytometry, by different kind of microscopy techniques, also by confocal microscopy. We label the CACO2 cells, the nuclei by DAPI blue staining, and also the tight junction, the proteins in the tight junction by fluorescent antibodies. And then we saw this kind of feature, photographs. So you can see how this is a lactobacillus, but it's just a section. And you can see, you can see they are attached to dendritic cells and how the dendrites are clearly seen. But also here you can see how this interaction between the bacterial cells and dendritic cells are very, very, very specific. In fact, are very specific cell to cell. This kind of interactions. And in this kind of model, in confocal model, you can see here the nuclei of the CACO2 cells, the tight junction proteins labeled with this fluorescent, and you can see that the bacteria are always attached, these bacteria are always attached to dendritic cells in the basolateral compartment. This was the PhD thesis of one of our PhD students, what presented in the year 2006. But later, we wanted to know if this that we saw in vitro models happen in real life. And the first was to study this in a mice model. So we have this plasmid, PTG262, with a clonophenicol resistant gene, and we clone a DNA fragment that is artificial. It doesn't exist in nature. So we clone this piece in this vector. We transformed bacterial strains isolated from human milk. We gave, during the 15 days previous to delivery, 10 to 8 colony-forming units. And then this was, at that time, this was a problem. You know, we were worried about the transformation of the strain, this kind of thing, but we didn't think about how to milk mice. So it was a period of brainstorming in the lab. Any kind of thing. Of course, at the end of the day, we pick mammary biopsies just to avoid the potential contamination coming from feces of those animals. And then we plate them, this material, in clonophenicol containing plates, and by PCR, we could detect this fragment in the mice of this group, but not in the control group, that they were very close to each other. More recently, we have used bidimensional in vivo imaging systems using the Lux operon just to follow this bacteria. We also transformed 2 species of lactic acid bacteria, and we check that they were bioluminescent upon transformation. This is a paper that was published last year. And then we found that just immediately after inoculation, you find here a big stain in the stomach, of course, but then this signal disappears, and you find bacteria in other places inside the body. But, of course, you can also find them in deep mammary biopsies, these luminescent bacteria. And, of course, you find them in the milk when this is collected. So, finally, we wanted to know if this happens in women. So, of course, we cannot use genetically labeled bacteria. So we use 2 different strains, one Lactobacillus gasseri strain and one Lactobacillus salivarius strain, both isolated from human milk. We gave to lactating women, and then initially we checked the presence by colony hybridation using a Lactobacillus salivarius probe and a Lactobacillus gasseri probe, and we, of course, found that some of the bacteria present in milk belong to this species. And later, by pulse field gel electrophoresis, we found that the bacteria we gave by oral route, in this case, the Lactobacillus salivarius, was also present in the milk, and also the Lactobacillus gasseri strain was also present in the milk, of course, at low concentration. In the year 2005, I was invited by Eduardo Schifrin, working then in Nestlé, because he was intrigued. It sounds interesting to him, but don't believe at all. So I came for a talk, and then he told me, I don't trust too much but I'm going to check. So I'm going to check, and they do their own assays, and they show sharing of bacteria from mother, all the samples. This slide has been shown before, is the same on this paper. They show this not only for bifidobacteria, but also for Staphylococcus epidermidis and also for Streptococcus salivarius, so that there might be a physiological bacterial translocation from gut to the mesenteric lymph node, and this thing, and was published in the year 2007. Of course, we know that there is incredible changes from fertilization to delivery at the embryo and the fetus level, but of course, parallel, there are a lot of incredible changes in women, just adaptation of every tissue, every organ, every system for the pregnancy and for future lactation. And this involves, for example, the gastrointestinal tract, but from the mouth to the colon. Also, the vascular system, the cardiovascular system. Of course, the immune system. Also, the uterus. And of course, the mammary glands. And all this adaptation, without no exception, favors a physiological translocation during this stage of life. And this doesn't mean an indiscriminate translocation as enteropathogens do, that broken all the gut epithelium. It's just a delicate and selective and I think, very complex pathway involving complex interaction between microbes, between epithelial cells, between immune cells, which are unknown at present, or largely unknown at present. But just, for example, you can see here the mammary gland of a non-pregnant woman and the mammary gland of a woman at 8 months of pregnancy. The structure is completely different because of huge angiogenesis process that are taken inside the mammary gland. So it's a completely different structure. You can see here endothelial cells. You can see red cells, how they are here. So very, very, very close to the mammary ducts. So translocation is not an unusual thing. It's a thing that happens within us every day, but just at very, very, very low rate. And for example, this is a work from a Cork group. They developed a system to monitor the presence of Bifidobacteria breve in mice in vivo, and they found, and it has been repeatedly found in other labs, that, for example, Bifidobacteria, we don't know why, they go to tumors, to solid tumors, and solid tumors are very enriched in Bifidobacteria. And let me just read the summary of a series of articles for this group. It's oral administration of the commensal non-pathogenic B. breve, this strain, to mice resulted in their translocation from the gastrointestinal tract with subsequent homing to and replication specifically in tumors at levels similar to intravenous administration. Reported genes were visible for at least 2 weeks in tumors. Mice remain healthy through all the experiments. Cytokine analysis indicated a significant upregulation of interferon gamma in the gastrointestinal tract of Bifidobacteria mice, which is associated with increase in epithelial permeability. BBD feeding did not increase systemic levels of other commensal bacteria. Presence of tumor was not necessary for translocation to systemic organs to occur. And these findings indicate potential for safe and efficient gene-based treatment, detection of tumors, this kind of stuff. And this is not exclusive for Bifidobacteria. You know, this probably the oldest probiotic strain, E. coli Nissel 1917, and this work show that a genetically engineered Nissel strain with a LAC-SET gene is the most sensitive way to detect hepatic metastasis because of the ability to translocate to the liver, to be enriched in metastasis, and after a clip of a certain substrate, then give a yellow color to the urine. So Oral delivery could lead to the preferential colon of tumors, and this phenomenon has been attributed to at least 3 different factors, because of suppressed immune surveillance with the tumor, because of the tumor huge vascularization, and also because of an increased ability of nutrients in the necrotic tumor core. So just if we think the same factor are present in the mammary gland during late pregnancy and during lactation. So there is a physiological immunodepression state, there is a huge angiogenesis process going on, and there is a lot of nutrients for this kind of bacteria. So these are conditions that may favor homing of bacteria to the mammary gland during these stages. Of course, I have talked about the potential origin from the mother, but of course there are bacteria that are not coming from the mother or not for the family. For example, maternal and family sources of bacteria to the milk usually involve gram-positive bacteria, 10 to 2, 10 to 3, relatively small amounts. But for example, if a mom is pumping the milk is risen with water, and water contains microbes, usually at a higher concentration than naturally present in milk. And usually there are gram-positive bacteria, yeast, and a very high concentration, up to 10 to 3 colony-forming units per milliliter. Also poor hygiene during milk collection. So I mean that to study the human milk microbiota is also very important, the sampling procedure, so because it also can mislead in this kind of studies. So now for the next 2 hours, I want, just the second part is about the potential uses of the knowledge on the human microbiome. And then I must say, I think something that has been told before here, that when we are studying milk as a whole, it's very difficult to distinguish between the roles of bacteria and the roles of other milk components, because most of the time there are a lot of synergistic actions difficult to separate, to differentiate. So just an example, there are many compounds with antimicrobial activity and other kind of activity. This is just a slide showing the same, just focus on 2 components, the human milk oligosaccharides and bacteria, how they can share different roles in the same baby. And of course, we know that there are many factors that may modulate the milk microbiota: host factor, microbial factor, environmental factors, socioeconomic factors, medical factors, anything you can have in mind. Some of them, very few of them, we know something about that. Most of them, we don't know what the relevance. So any kind of thing, you can think about it. But at the end of the day, women with different milk microbiome profiles may have a normal milk microbiome. It's something like happening in the gut. So it's very difficult to define a normal gut microbiota only because of the names of the microbes that we have there. So if the composition may not serve to define a healthy human milk microbiome, how can we define a healthy human milk microbiome? I guess that the answer is in the roles. Which roles we expect from a healthy human milk microbiome? This is more than 1 century ago, but I think it's the same. And of course, we can isolate, and we have a huge collection of strains from human milk, from different species, from different genus, and we know that they can have several properties. But of course, again, these properties are at the strain level. And we have genome analysis, or we have a genome sequence for many of these strains, and we can find very, at least theoretically, relatively nice roles. And you know, when we talk about the human milk bacteria and the roles for babies, we usually think in the babies. And of course, we also do. We are studying the roles maybe for preterm babies, in HIV mother-to-infant transfer, and also in some respiratory tract disease like bronchiolitis by respiratory syncytial virus and these kind of things. However, sorry, I just want to use one example that is focused on the mother, because usually mothers are just forgotten. In fact, the breast during lactation is a kind of orphan organ because nobody studies when there is health problems at this stage. It's an organ that science has not go inside, and I think is required. So when in normal condition, in healthy conditions, we usually see in human milk samples a high diversity, but at a low concentration. However, when there is, for example, mastitis, we can see that diversity is very, very, very reduced, but the concentration is high, is up to 10 to 6 colony-forming units per mil. This means that a mammary duct is filled up with bacteria. There's no room for a single cell more. And in these cases, some Staphylococcus, Streptococcus are the main cause of this kind of problems. And we can see this by traditional culture methods. For example, here is a healthy sample, is undiluted milk, and you can see some Staphylococcus in this sample. But, for example, look this case of acute mastitis and subacute mastitis, that the milk diluted 1,000 times contains the same bacteria that undiluted samples in a healthy host. Also, we can see by optical microscopy, by normal traditional microscopic techniques. Here you can see, in a case of mastitis, how there is a large number of staph. There are also T cells, there are epithelial cells, because of course there is an inflammation. And in the previous talk, they were talking about EGA coated, but you find, although you cannot see here, but all these bacteria are full of IgA around. We don't know why, we don't know for, but this is the fact. They are coated with these antibodies. And then this is, for example, a preparation from healthy breast. You can see bacteria, but of course the number is much lower. Of course, we also can see by microbiome-related techniques. For example, this is from Maguire lab, a case of a recurrent mastitis. So when there is mastitis, the diversity is almost lost. And then when recovering, the diversity is increasing. When a woman is feeling well, is maximal, the diversity, but again, is lost, and then you have another episode of mastitis. Recent reports has also shown that the confirmed previous hypothesis that mastitis women have lower bacterial diversity, increased abundance of opportunistic pathogens, and depletion of commercial obligates and anaerobes. So mastitis, I am afraid that human mastitis is a kind of iceberg. I mean, most of the cases that are diagnosed are because they are acute mastitis with clear clinical symptoms, fever, breast redness, this kind of stuff. But most of the cases belong to this part of the iceberg. And then acute and subacute, chronic are not arbitrary terms, are terms that are proposed by the National Library of Medicine, and this part of the iceberg is essential to understand the etiology, pathogenesis of mastitis, to have a better diagnosis, but also to improve treatments when there is a painful breastfeeding or lactation. So when we have acute mastitis, most of the cases caused by Staphylococcus aureus strain. Of course, there is an infection, the proliferation. This Staph aureus produce from one to several toxins in the mammary tissue. Of course, these toxins are responsible for the acute inflammation, and the breast is so hugely vascularized, then most of the toxins are absorbed. They go to the bloodstream, and then you get systemic symptoms like a flu-like disease. And of course, you can see here, but of course, in some cases, not only aureus, also other bacteria, but some cases, 2-3% of cases, they might lead to a breast abscess. However, this part here, the hidden part of the iceberg, usually are caused by coagulase negative staph or strep. What you find in milk is always contamination, but it's not the same to have 100 than to have 1 million per milliliter. Of course, it's different. So the beginning is the same, But the thing is that when they increase inside the mammary duct, they have a huge ability for biofilm formation and also a huge ability for antibiotic resistance, not only because of the presence of antibiotic-resistant genes, but also because they form this biofilm that is a form of antimicrobial resistance itself. So it usually become recurrent chronic infections. There is no toxin, so there is no breast redness, there is no flu-like symptoms, there is no fever, or very, very rare. But this is why they are widely underrated, underdiagnosed. So in this case, that is painful breastfeeding, well, this might be the normal way. It's not the normal way. You have a high concentration of this kind of bacteria, and the concentration should be much smaller. So this woman has not breast redness, but has pain, needle-like pain, burning pain, and has engorgement in the breast. So biofilms are a key player in the pathogenesis of this kind of conditions. And you may say to me that Staphylococcus epidermidis is a commensal species, and you are right. And not only a commensal species, sometimes can be a probiotic species. There are strains of Staphylococcus epidermidis that can compete with aureus, with Streptococcus pyogenes, with other microbes that are pathogenic. So they might even play probiotic-like roles. However, we also know that Staphylococcus epidermidis is one of the most important opportunistic pathogens, not only catheter infection, but, for example, in pyelonephritis or osteomyelitis or this kind of conditions. So the fact that behave as a commensal or a pathogenic strain may depend on the host, the ecology, the strain, the properties of the strain, and also, of course, the concentration. You can see here, this is a biopsy of a mammary gland of a healthy woman without no problem at all, and you can find bacteria, you can find cocci, you can find small rods, but a very small concentration. And of course, you can see here them just attached to the fat globules of milk, where they tend to concentrate. But just look to this picture. It's a mammary duct with a Staphylococcus epidermidis mastitis. It's a huge biofilm, a lot of exopolysaccharides, a lot of exoproteins, all this kind of stuff. So this is a form of resistance to the host immune system, and also is a way to resist antimicrobials, for just as an example. So, of course, if some of these strains that are resistant, some resistant strain we cultivate in the lab, we add a beta-lactam antibiotic to the growth medium, they grow even better. They can use the antibiotic as a carbon source. So when the other sources are gone, of course, by microscopic electronic microscope, you can see it's like very, very beautiful pitches, no? Like. Of course, when we isolate sensitive bacteria, they are destroyed by the antimicrobial, and also so we know that sometimes we found bacteria that are resistant to almost everything, just because the genes or because biofilm formation. So then you arrive to these kind of situations. This is one of the pictures that I did that I like the most, because what do you do here? You has to come an helicopter or something like that. So, and this happens. So we have to check for alternatives, and the one that we have at hand at that time was the use of probiotics. So very rapidly, we use the proposed frame from the World Health Organization and the FAO to select and to prove. We did one first a clinical trial, a small clinical trial with a placebo. We found statistically significant differences. We were told by the pediatricians, midwives, to please continue with this clinical trial. We did a second trial. The ethical committee at that time didn't allow us to have a placebo group because of the results of the previous trial. So they told us to include a control group in which antibiotics given in normal practice were given to women with these problems. So we found large differences before treatment and after treatment, both in the microbial count and also in the feeling of pain of women. So any one of you want to know more, there is the Reuter company, it's produce a small video, 3 minutes. In 3 minutes, they explain better than me in half-life, so I don't know why, but it's true. So later, we did a prevention study during pregnancy, and the results also were quite nice. We have checked for kind of markers. Of course, we can find markers in total bacterial counts, in somatic cells counts, in polymorphonuclear leukocytes, and these kind of things. But we also check for markers in the urine, metabolomic markers. And it was a double-blind trial. But at the end of the study, we found that before the treatment and after the probiotic intake were completely different response. I mean, here you find a lot of lactose because the permeability of the mammary epithelium is lost, and then you find lactose in urine, but when the integrity of the epithelium is established again, then there is no lactose in the urine, so it's a good marker for this kind of a rapid marker. We also did transcriptomics from the cells, the blood cells, and also the milk cells. And in the case of the milk cells, because in the blood cells we don't find nothing statistically different, but in the milk somatic cells, we found that initially we found an overexpression of pathways involved in cell death, apoptosis, inflammation, these kind of things, while after the treatment, there is an overexpression of pathways involved in normal cell processes, secretion systems, this kind of stuff. This has been published a few months ago. So just to finish, mastitis can be relatively common, unexpected, frustrating, hard experience, but is just consider a kind of second, third-class infection, maybe because it's just women during lactation, so they have two options. Don't complain at all, just keep breastfeeding sometimes against the opinion of the pediatrician or even the family, or just giving up a lactation. You have a third way, that is just healing, or try to heal. So this is quite nice cartoon from Western University of Australia. Mastitis is the first medical reason to stop breastfeeding earlier than expected. And we know every day is more clear the connection with our health when we are born and our health when we are elderly. So I think mastitis is not a lifestyle, this kind of thing. It's a public health issue. Just to finish, I think one of my latest strengths, I like to present here in this slide our present solid knowledge on the mammary, the human milk microbiota. So that is not too much. We start to know just small droplets, but the picture is almost empty. So there is a need of a lot of research. I like very much also this graffiti in Madrid in a wall. In the past, I have some doubts. Now I don't know. Maybe it can define my present situation, scientific situation. So it's a lot of research is required to clarify this point. Here there are immunologists, I think from also neurologists, biochemistry, biophysics are all required to come to this field. So people who are now at present working in the lab, and thank you very much for your attention.
We have some times for questions. Jerry.
Very comprehensive lecture, too many things to say. But anyway, and congratulations to the Rubens's painting of Origin of Milky Way, which you showed us. How to collect murine milk is relatively simple. You cannot squeeze the gland and so on, but you let the pups drink the milk, take the pups, and they will have white spot on the stomach, and you can aspirate it. So you can.
Yes, we did it. We did it. We did it in the three ways. We collected milk samples directly from the mice, but also we have this problem that maybe fecal contamination can change the data. So we collect milk from the gastric content of the pups, and also we perform mammary biopsies of the mice.
And the second concept, a very small proportion of children are born who secrete milk, so-called witch's milk. No IgA in it, nothing. Have you ever looked at the microbiome of the witch's milk?
No. We are looking forward to do that. You know when you are working on human milk microbiome, you find a lot of strange things that you never think about that. Because I mean there is 2-3 days old children
they
with
secrete
mastitis.
milk for 2-3 days.
Or you find 90 years old women with mastitis, and you find microbes inside. And this is not unexpected. But when you are working on this kind of thing, sometimes the hospital says, well, I'm going to send you this person because I don't know what's happening. And you find bacteria inside in this period. And for example, when there is sometimes that some women has problem in the breast and is coincided with a prolactin peak, sometimes at that time you can find bacteria inside.
Okay.
So,
Are there any other questions?
please, other questions?
Very comprehensive lecture. If not, I think we will discontinue. Thank you very much.
Thank you very much.
It's a real pleasure for me to introduce Dr. Pia Panaraja, who is an associate professor in pediatrics and molecular microbiology and immunology. And she's going to talk about a slightly different topic today, and that is the virology of breast milk.
Hello, everyone. All right, I'd like to thank the Nestle for having me and all the chairs for inviting me here to talk today. As Dr. Walker said, my talk is a little bit different. We've been talking a lot about the bacteria, and so today I'm going to talk about the viruses, or the virome. Okay, so, and if that blank slide of what we don't know about the milk, you know, the virome has even less data. Okay. So viruses actually comprise the highest number of species on Earth, or highest number of different particles. So 10 to the 31 different viral particles exist on Earth. And in just 1 ml of seawater, you can find 10 to the 7th viruses. That's really a crazy number if you just think about that. And so viruses, of course, because they're everywhere on Earth, we are going to find them also on the human body. So today I'll talk a bit about the virome of human milk and infant gut, the very few studies that exist out there. Talk about virome transmission with the focus on mother to infant, and then describe the role of the virome on infant health. And so when we think about the infant gut, this is the gut here, we've been mostly concentrating on the bacteria. But in addition to the bacteria, we have these eukaryotic viruses that live there, and then the bacteriophages. And they all play very important roles. So most of the time when you think about human viruses, you think about transient viral infections. These are things like rhinovirus or coronavirus that cause the common cold. There's lots of these viruses out there. And then there are the persistent or latent viruses. These are in the herpes family, for example, that can cause cold sores that they persist in your body and come back every time your immune system is a little bit depressed. And then there's endogenous retroviruses, and these actually make up about 8% of the human DNA. And they are incorporated into the human DNA, and then actually can even be incorporated into the germ cells and get passed from generation to next generation. And then there are the bacteriophages. These are viruses that infect bacteria, and they comprise they are about 10 to 11 bacteriophages per gram of stool. And then, in addition to all of that, the majority of the viruses that we identify when we study the virome are actually novel, unidentified viruses that we have no idea about yet. So here are some pictures of bacteriophages. And these are bacteriophages that are attached to a bacteria. You could see there's just so many of them here, and again here in yellow attached to this bacterial cell. And so why do we care about these bacteriophages? They can attach, they have these cycles, so they are here attached to a cell and can undergo two different lifestyles. So one is they attach to the cell, insert their DNA, and then the DNA, this hijacks the bacterial replication system, produces more of these bacteriophages, and then eventually lyse, and you produce more phages. And so in this manner, though, they also kill the bacteria. The other important life cycle that they have is the lysogenic cycle. So here, the DNA is inserted into the bacteria. Then that inserts into the host gene, the DNA, and then that can actually induce changes in the host bacteria so that it can actually be more fit and actually perhaps replicate more. Or it could also induce antibiotic resistance genes. So there are some advantages and potential disadvantages of this happening. And so this is why we care, because the phage and bacteriophage are the same thing. These phages can impact human health. So the Shiga toxin and cholera toxin, for example, are from phages. They can also determine virulence and pathogenicity, can bring about antibiotic resistance, and actually change metabolic capacity. Oops. And I put this here to remind me the German outbreak about maybe close to 10 years, between 5 to 10 years ago, that was an E. coli, a Shiga-producing E. coli, Shiga toxin-producing E. coli that were decidedly from sprouts that affected nearly 4,000 people. That actually started with a phage, and then it was transferred actually from one E. coli to another E. coli, so bacteria to bacteria transfer that then increased its pathogenicity and virulence. And in addition, there's even some, there's phages responsible for fluoroquinolone resistance to those same Shiga toxin-producing bacteria. So just an example of how these phages impact our life. So before I go into a little bit more, I just want to show how difficult it is to study the virome. This is the everything that's involved. When we have either stool or breast milk, there's a lot of filtering involved to filter out cells, cellular debris, to get rid of human DNA that's there. And so it's before the sequencing. So it's not quite as easy as studying the bacteria. And that's partly why there are so few studies out there, because of the difficulty and the challenges, and also because of the cost. So these are the studies that have looked at the virome. It's actually not a complete list, so it seems like a lot. Some of these, however, have been using PCR studying, using specific viral primers and primers for bacteriophages, while others have looked at sequencing. But you could see that it's really been looked at many different sites, very similar to the bacterial sites that was just presented. So oral, gut, blood, urine, CSF, that actually just came out earlier this month. Multiple studies in the respiratory tract, only a few studies in milk. a few studies in amniotic fluid, one of which showed that there was there were no significant viruses in the amniotic fluid, and then skin. And these are all the different types of viruses that are there. And so there are many, but what we'll soon get into a little bit more is that a lot of these are bacteriophages. So we decided to look at the milk virome. We wondered if it was different from other body sites. And so you could see here, this is the adult milk in blue, and infant feces in red here. And then they separate out from the adult urine, adult feces, and adult saliva. So they are it's a different community in the milk and in the infant gut compared to the other body sites. In addition, it seems like the milk and the infant gut tend to cluster closer together compared to the other adult body sites. And when we look at mother and infant stool together, mother, milk, and infant stool, a lot of them cluster together, but there are a couple that cluster a little bit separately. So we wondered, and then when we look at what was there, you could see here, these are the eukaryotic viruses, and then the bacteriophages, and over 95% were bacteriophages, both in the milk and the stool. And so we wondered if, and these are the top three there, the myoviridae, doesn't like me right now, pushing it too fast, okay. The myoviridae, cephoviridae, and podoviridae, and they're really the differences are the tail of the phages. And then when we look at diversity, the viral diversity tends to correlate with the bacterial diversity. So stool, this is infant stool, and these are infants that are less than 1 month of age, so very young infants. The stool viral diversity here is actually still quite low, where milk viral diversity is higher. And this is the bacteria. Again, the milk is higher in diversity than the infant stool. And so this actually is similar to adult data, where they've looked, this was from a twin study, and they looked at bacteria diversity, and there were some where the twins had similar diversity, and some where they were discordant, shown here. And the viral, here on the x-axis is the microbiome, so the bacteria alpha diversity, and then here is the viral alpha diversity. And what we see is that as you increase the microbial biodiversity, you also increase the viral biodiversity. But interestingly, this is only seen with the bacteriophages and not with the eukaryotic viruses. So the bacteriophages, which infect the bacteria, go up in diversity with the number of bacteria, but the eukaryotic viruses are pretty stable without dependence on the microbiome diversity. So these phages, remember I talked a little bit earlier about the three most common phages that we saw, have these different tails, and that will determine how it attaches to the different bacteria and who it can attach to. So, for example, these are the different attachment, the specific bacterial receptors that are found in gram-positive organisms. And then here in the middle here, found in gram-negative, in both organisms, and then down here, gram-negatives. And so depending on the type of tail, it can look for certain receptors in order to be able to insert its DNA. And so this is important because that will determine what type of bacteria it can infect. So, virome transmission factors. So there's a lot of different ways that viromes can be transmitted. And so there's some that are modifiable, some that are fixed, and then others that are potentially modifiable. So age, sex, genetics, that's all fixed. But people have looked at differences in adult gut microbiome and also in blood, and these were all affected by age, sex, and genetics. And these are the potentially modifiable factors. So potentially, delivery method sometimes is modifiable. We're definitely seeing a lot more cesarean deliveries than we used to, and so some of that may be some people may have an elective C-section just because it's easier. But that also, in addition to changing the microbiome for the infant, that also changes the virome. The environment affects the viruses that you have on your body. Household contacts, there's been a lot of studies of people adults living together. They tend to share viruses for a period of time, then it goes away, and then somebody may bring in a new virus from the outside, and then they share it again, and it goes away. Antibiotic use also affects the virome, most likely because it affects the microbiome, and then therefore the bacteriophages might go down in number and diversity. And then diet, breast milk. So very early in life, the first thing that happens is birth, right? So as I talked about just a little bit earlier, during pregnancy, they didn't seem to find a lot of significant numbers of viruses in the amniotic fluid. So then the next thing that happens is birth. And whether you're delivered by vaginal or C-section, you could see, this is the beta diversity of the babies born via C-section compared to vaginal, and they do cluster distinctly. And these are the different types of viruses that are there, and again, differ by C-section versus vaginal delivery. So that's an important factor. The next is breastfeeding. So this was actually shown earlier today, but just like we looked at in breastfeeding with bacteria, we saw that in bacteria, if you're exclusively breastfed during the first month of life, the moms and babies actually share about 35% of the microbiome, the bacteria microbiome. And then that decreases over time, which makes sense as the baby starts to feed from other means, the introduction of solid foods, and also gets around in their environment, crawling on the floor, putting things in their mouth. That changes the bacteria flora. And so we wondered the same for, and so we think that a lot of that is mom to baby, but there's probably a component of baby to mom, and it just sort of cycles between mom and baby. And so we asked the same question for viruses. So do the viruses come from mom's milk, or could it be the same as other moms, or just kind of random from the environment? And we know that viruses can come from milk. There were some hints earlier with the mom's milk, the communities being more similar to the baby's milk. We also know that there are viruses such as CMV or HIV that transfer from mom's milk to the baby and can infect the baby. But what we didn't know as much about are the bacteriophages, which comprise 95% of the viruses that are there. So this was the first study that in 2008 that looked at both phages and bacteria that transferred from milk to baby. And so they found that there were phages that infect lactobacillus, lactococcus, streptococcus, and enterococcus, species that were all dominant and abundant in milk. But they did not actually find the viral sequences of the phage in the milk itself. But the method using here was using primers against some bacteriophages of significance. and not looking at all of the virome. So, and then this paper, which came out in 2017, looked at the maternal inheritance of bifidobacteria communities and bifidophages. So they focused in specifically on the bifidobacteria and their phages. And they actually showed that there was vertical transmission. So then they sequenced the bifidobacteria in milk and the bifidobacteria in the baby. They found that they were the same, and they found also that the phages were the same. And what they found, though, was that the phage was transmitted as a prophage. So in the bacteria, it had already integrated into the host gene. It was transmitted as a prophage, and then in the baby gut, actually was able to replicate and become more phages, leading to cell lysis, but having lots more phages to go around to all of the other bifidobacteria. And then in our study, we also looked at homologs within pair. So again, mom-baby pairs together, how much of their sequences were the same compared to baby with a random mother pair. And so you could see if we, within pair, the numbers were always higher than if we paired that baby randomly with another mother. These were the viruses that we found in the three pairs that had the most homology. And I really put this up there just to show that there's a lot of homology, but also that the significant amount of what was shared were bacteriophages. So the question is then, once these phages and other viruses get to the gut, what happens to them? How long are they there? And unfortunately, there's not a lot of data about this in pediatrics, but in adult data, this was a study of one adult man grabbing stool at multiple time points, and they looked at how different those communities were by the interval between time points. So this is only 1 day, 10 days, 100 days, 850 days. And you could see, and so then the higher the number, the less divergent, and the lower the number, the more divergent. So here, if it was only a few days in between samples, you didn't see a lot of change. And then as you increase to the large number of days, 850 days, so 2.5 years, then we see more changes. And the colors, actually, I don't know how well the colors show up for you, but the temperate viruses in blue are the ones that are more often transmitted as prophages or are integrated into the bacterial gene. And you could see that there's less change for them compared to the not temperate viruses. And on your right-hand side, we look at the viral families and the substitution rate. So as the DNA replicates on itself, then that's occasionally there will be substitutions into that, and that could lead to new genes, new antibiotic resistance, et cetera. And so the microvertiae had the highest amount of substitution. It was about 4% in this particular study. And that it was suggested that it's possible because that's a single-stranded DNA phage compared to the rest, which are double-stranded. And so with the single-stranded, it's a lot more error-prone compared to the double-stranded phages. And then phages then can move between individuals. Now this study is a study of fecal transplant. So it's not at all the normal way that phages move between individuals. But I think it's still interesting. It's three pediatric patients who had ulcerative colitis, and they were treated by stool transplant actually between 22 and 32 times. So they receive a fecal microbial transplant 22 to 30 times, which seems like a lot for me. But anyway, it's from a single donor. And so on this side, on your left, you have the patient, their original virome, and then your donor virome just below them. And then the first column is pre-transplant, then during the transplant, and then post-transplant. And what you could see in pink here, a lot of the pink had the largest amount that stayed around post-transplant in all three of them, and that was the Saphoviridae, which is interesting to me because that was also one of the highest amounts, one of the highest abundance for the phages in our studies when we looked at maternal milk going to the infant gut. The Saphoviridae was also one of the highest that transferred and then stuck around for a little while. So these are the only studies that actually looked at milk and infant gut samples over time. And what they found was that this is the bacteriophages, and then this is the eukaryotic viruses. So the phages actually start out quite high and then decrease over time. And then the eukaryotic viruses actually start low and increase over time. And that makes sense to us as pediatricians because the kids will encounter different viruses in their young life, and then some of those persist in the body over time, and so that has a slow, gradual increase over time. But these bacteriophages go down, and this study compared bacteriophages in orange here and the bacteria in blue. And so you could see that the bacteria, as the bacteriophage goes up, the bacteria comes down. And same here, that basically there's this predator-prey relationship between the bacteriophage and the bacteria. And some people call it kill the winner. The predator-prey suggests that there's some cycling up and down of the different, of the phage and the bacteria. So here is depicted here. We have the phage here kind of going down, but cycling a little bit where the bacteria go up in number. And then in blue, down here are the eukaryotic viruses. And this all tends to stabilize over time, similar to the bacteria, around 3 years of age. But this whole middle section with the dotted lines are really a question mark because we don't have studies we don't have enough longitudinal studies with enough numbers to really know if this is true. This is all hypothesized based on the 2 studies that you saw earlier on what happens with the phage and the bacteria. And so one wonders, with this change over time and with the number of bacteria going up and down, if any of those kind of cross these windows of opportunity where there's some critical time periods and time points in early life where the microbes, particularly the bacteria, influence immune development and metabolic development. And so if the phages are there, then perhaps they're also helping to control the bacteria so that there's not too big of a proliferation of the pathogenic bacteria or the ones that don't induce immune tolerance correctly. And it's interesting to me that Mother Nature has sort of introduced all these different factors perhaps to control the bacteria, the potential for IgA and an antibody in milk to bind to certain bacteria, potentially pathogenic bacteria, so that they're not misbehaving in the infant gut. There's the potential for oligosaccharides to act as prebiotics, to feed certain bacteria, to favor certain bacteria over others. And then there's the potential now for viruses also to either infect and kill off bacteria or to introduce the good genes in them to make them more fit in their environment. And so this is one study that just came out recently that showed whole fecal metagenomes and viromes, where they looked at the potential for both bacteria and viruses to act, so different cellular functions that are possible. And so you could see up there some of the different processes, lots of cellular processes, metabolism, and then here specific KEGG pathways that are involved. And in there you see a lot of metabolism, infectious diseases, which also implies immunologic diseases or immunologic teaching, and many other processes. This is in adults, but still possibly transferable to pediatrics. It's something that needs to be studied. So now I'll present just the very few studies that have been done in pediatrics. So this is there was one group that looked at infants with diarrhea, and they classified them by the type of stool. So from pellet, so that's, I guess, maybe too dry, but maybe a good stool, all the way to watery, with several classifications in between. And so they looked specifically at the eukaryotic virome and saw that there were changes associated with the type of stool consistency. But it's really hard to know, was this the cause or the effect, or just an association, maybe neither, just an association. But these are so far, and you could see this was just published 2019, these are so far the only types of studies that are out there. Another study, it's a little bit out of order. Another study looked at autoimmune diseases and specifically type 1 diabetes, and found that the intestinal virome changes precede autoimmunity in type 1 diabetes in susceptible children. So again, hard to know cause and effect, really just starting with an association type of study. And then Jeff Gordon's group looked at malnutrition. So they looked at virome patterns in healthy and malnourished twin pairs from Malawi. And they found that virome patterns were concordant in healthy twin pairs. So if you have two healthy pairs, the viromes were very similar. But then if you had one twin became malnourished, then you also saw some similarities. Actually, both viromes became altered, and there was delayed gut microbiome development associated with it. You all probably remember he had their group has published extensively on the gut microbiome and showed that there's delayed gut microbiome development or maturity in children that are malnourished. And so what we saw there is that when they looked at the viruses, what they found was an increased prevalence of this virus family, Anelloviridae and Cercoviridae families in phage populations that could distinguish the malnourished twin from the healthy twin. And these two families have also been associated with increased diarrheal illnesses, and also with looking at adult HIV-infected versus not. The HIV-infected also had an increased prevalence of this Anelloviridae family. And then another group in China followed around 4,500 children over a respiratory season, over several respiratory seasons, and looked at how many times they developed acute respiratory tract infections. And so what they found, they looked at all of the virome, but they focused in on this Propionibacteria phage, and they found that kids that develop multiple acute respiratory tract infections had higher amounts of this phage, significantly higher amounts of the propionic bacteriophage compared to those that only had a single acute respiratory infection. And here you could see this, it goes up with the actual number of respiratory infection, first, second, third. And what they found was this propionic bacteria actually had the ability to induce certain increased amounts of cytokines in the respiratory tract, perhaps leading to more symptoms, more inflammation in those children with multiple respiratory infections. Oh, and then this study also just came out earlier this year. In Bangladesh, they looked at children who were vaccinated with polio vaccine, and then looked at immune response. So in the lighter color here, seronegative, in the darker color, seropositive, meaning that they converted and had increased antibody. And they looked at the amount of the prevalence of other enteroviruses. So the question was, could other enteroviruses interfere with vaccine response to poliovirus? And what they saw was yes. So you could see in the seronegative group, again in the lighter color, they all had higher amounts of each of these enteroviruses in their gut at the time of vaccination, and they did not respond, and so they did not respond as well to the vaccine. So it's an interesting concept that these viruses can also interfere with antibody response and affect the immune system. So those are all the studies that were done in children. So again, not very many, but one can kind of think about, potential next steps and potential uses for viruses if we were to find them to be helpful. So here, kind of nicely documented in a figure, potential phage-inspired approaches. So you could give phage therapy where it, mostly attacks the pathogen, the pathogenic bacteria, and so then they all die off and you're left with some good bacteria. You could have phage enzymes kind of similarly doing the same thing, biofilm dispersal by the same method, drug sensitization. So you can kind of introduce phages to either, again, promote the bacteria that are present or kill off the pathogenic bacteria that you don't want to be there. But there's still a ways to go. There's a lot of limitations of the current studies, and most of the studies that we know of so far are cross-sectional data with a small number of subjects. And even in the few that were longitudinal, again, a very small number of subjects where we have data over time, especially in early life where there are so many changes to the baby. And there's a lot of studies where they only look at the baby, but it doesn't include mother and infant as a pair, so we could actually study the milk microbes and viruses and the baby's microbe and viruses. And the technology is still expensive. It misses a lot of latent viruses, integrated viruses, double-stranded DNA viruses, and then large eukaryotic viruses. So there's still a lot of unknown. These actually, in several studies, can be up to 70, 80, even 90% of the viral particles that are there. So we really are just knocking at the very tip of the iceberg. And it wouldn't even be like the iceberg picture you showed earlier. It really would just be like a peck at the iceberg. And there's a lack of standardized protocols and incomplete viral databases. So that's the other part of the problem is that we use the viral databases that are out there, but because many of these viruses are novel and unknown, it's hard to tell exactly what they are. So kind of leave you a little bit of the slide of what we hypothesize happens, kind of a summary of some of the things that I talked about, that there are all of these different factors that influence the virome, both in mother's milk and in the infant gut. And there's this trajectory over time that's still unclear, where we need more data, but I think it's a nice potential field of study. And then just like with the bacteria, we need more mechanistic data of what actually happens to try to move beyond the association data. So with that, I'd like to thank, these are lots of students that work with me, my former mentor, some collaborators. We're extending our studies. So I am actually a pediatric infectious disease doctor and by training more of a vaccinologist. So I'm very interested in immune responses to vaccines. So we are looking at, and so there are some vaccines that have significantly different rates of response in the developed countries compared to developing countries. And so we're trying to understand why, kind of focused on the immunologic aspects, and then trying to understand how the viruses and the microbiome play a role in that. And so these are collaborators there. And then at UCSD, David Pride, who I worked with on our virome studies, the Rick Bushman, University of Pennsylvania. When I first started on the microbiome, I went over there and spent just a couple days in his lab talking with the wet lab people and the bioinformatics people to kind of learn all of this. So thank you very much.
This is now open for discussion.
Go ahead. Can I ask a question? Yeah. Okay. Thank you very much for your nice presentation. As you have mentioned, a study from Bangladesh referring to the immunogenicity of polio virus. So we know, by and large, when a phage infects bacteria, so they multiply inside the bacteria, okay, and ultimately the bacteria dies, and the viral population increases. So by law, there should be more immune response if there is more bacteria which is infected by the polio virus, and the bacteria dies and the virus flourishes up. So there should be more immunogenicity, and antibodies should be more in those cases. I mean, I'm contradicting the thing you have presented. This is one thing. Second, I like to a second question. That is, you have mentioned that persistent latent viruses. So you have included human herpes virus type 6. We know the human herpes virus type 6 causes roseola or erythema infectiosum. So it's a disease of the babies between 6 months to 2 years of age. They have a high grade of fever for 4 to 5 days. Then suddenly fever goes by lysis and the rash appears. When the rash appears, the baby becomes all right. And I do not know how these bacteria, why these bacteria remain latent or persistent, and how they present later life. Like your herpes simplex virus, type 1 and type 2, they hide in the somatic nerve endings, and the herpes zoster virus in the posterior root ganglia or in the cranial nerves ganglia, and they flourish up as shingles in later on life. But how the herpes virus type 6, where this survives, and what is the late presentation if they are latent or persistent? And I don't know the herpes simplex virus type 7, what type of illness they cause. I do not know. If you know, you can acknowledge.
Yeah. Okay. So the two questions. So one, the first question on the viral shedding and the vaccine response. This study focused mostly on the eukaryotic viruses and the enteroviruses specifically. So not on the bacteriophages. And so it wasn't looking at phages that would infect the bacteria and then lyse them. So I think that...
So it differs in prokaryotic and eukaryotic virus?
Yes, differences between eukaryotic viruses and bacteriophages. Yes,
okay.
so it's the bacteriophages that will affect the different bacteria that could either cause better fitness or lysis. And so I think that's what you're thinking about, that potentially if there's lysis, perhaps there would be a bigger immune response. But in here, it was that there were enteroviruses there, perhaps that may look somewhat similar to poliovirus, and so therefore they're vaccinating with poliovirus, and the immune system then thinks it already knows, how to respond to that particular virus, and so doesn't mount an immune response. Yeah. So, and then your second question about HHV6, so most of the time for us, it doesn't cause lots of problems later in life. When we see it is if you have somebody who's immunodepressed later in life, then the HHV6 will come. So in those with HIV, those who are immunocompromised from chemotherapy or mainly bone marrow transplant, so those types of patients.
Where they lie dormant? In which part of the body?
That's a good question. I don't know the answer to that. I don't know if any audience members know. Yeah. Yeah.
There are 3 viruses in humans of enormous medical importance associated with milk. HIV you mentioned briefly, and I would like to add that, of course, you can be infected that route, but it's relatively very rare. Mm-hmm. And as a matter of fact, WHO recommends for mothers who are HIV infected to continue breastfeeding because the probability of being infected with HIV is much lower than dying of diarrheal disease. Second, EBV. Epstein-Barr virus is present in human milk in relatively high proportion, and interestingly, children in Africa within 1 year of their life are all EBV infected. 96% of kids are seropositive for EBV, and apparently it comes from milk. This is not the case in Western Europe, where children are infected around time of adolescence, and in China, EBV infection 6 to 8 years. And again, there is a tremendous impact on that. I don't know how many of you are from Africa, if anybody is from Africa, but EBV, as you well know, causes infectious mononucleosis. Yes. It's a disease which is unknown in Africa due to the fact that kids are infected so early in their life and they mount without basically any perceptible clinical manifestation. They mount an immune response for lifetime, and they cannot be infected with EBV by the oral route and have infectious mononucleosis. And maybe you can say something else about cytomegalovirus, again, tremendously important virus, which goes before the birth or through the milk, and you know the consequences. So I think these are 3 viruses which really should be always reminded of us milk, because that's the way how they're transmitted.
Yes, thank you. Yeah, thank you for bringing up those very important points. I didn't choose to concentrate on those viruses because there's actually a lot more known about them, but I think I just want to reiterate your important point about HIV, that it can be transferred through milk, but is rare, and so the recommendation is to continue breastfeeding exclusively for 6 months and to a year if possible, and then to have a slow wean because they have found that if you wean rapidly, you actually have a higher risk of transmitting HIV from mother to infant. And then CMV, I know there's several neonatologists here in the audience, and so as you well know, there's lots of consequences potentially of CMV, particularly transferred in utero, but possibly also transferred to a premature infant. So yes, thank you for bringing that up.
Yes.
Hi, thank you for the wonderful talk. Vanessa Castagna from UC Davis. So I wonder if you could comment on bacteriophage therapy. This is known to be successful in eliminating bacterial infections. And what are your thoughts on utilizing bacteriophage therapy in modulating the microbiota in perhaps a dysbiotic state?
Yeah, that's a good question. I think that eventually that would be a nice therapeutic method. I think right now I feel like we don't know enough yet because there are so many different bacteriophages there, and we know that there are certain ones that are predisposed to infect certain bacteria, but it's hard to know if they also can potentially infect other bacteria, and so that perhaps you'd be doing a disservice if you're killing off good bacteria, killing off both good bacteria and bad bacteria. But I do see it as a potentially promising therapeutic in the future.
Yes.
Thank you very much. Nalini Jongriyapan, Mahidol University, Thailand. Thank you very much for your wonderful talk. You mentioned about diet, breast milk, and we all know that maternal diet affect nutrients in breast milk, and as well including prebiotics, right? That can affect probiotics or bacteria in gut, infant gut. What about the effect of maternal diet on breast milk with virus?
Yeah. I think that's a great question, but there's no answer that's known at this time. There just isn't a lot of data, or there isn't any data that I know of at this time to look at maternal diet and which viruses are in milk and how that affects the infant gut. But I imagine that that data will come out soon as there is more, as there are more people looking at the virome and learning from the past now, grabbing epidemiologic data as well. So hopefully we'll know soon.
Pia, there's a lot of parallels between bacteria and viruses. What happens when women are given antiviral medication? Does that disrupt the virus and its association with bacteria?
Yeah, that's a great question. I don't think there's any data out there for us to know for sure. Now, there are several antivirals that are out there, but they don't work very well. And two, there's very few. So acyclovir works very well for HSV, but not actually so well for maybe the rest of the herpes virus family. And not so well for other viruses, and same with the other antivirals that exist. And most of the time, they're not prescribed for routine viral illnesses. The only ones that are prescribed for routine illnesses would be acyclovir for herpes virus or oseltamivir for the flu virus. So I think we don't know yet what effect that might have on the other viruses, but I think maybe not as much as we might think, unlike antibiotics, just because our antivirals are not that effective in general.
Yeah. I have another question with regard to the Jeff Gordon studies, where they showed that children who are malnourished have an immature bacteria that persists to later in life. Does that same thing happen to viruses?
Yeah. So in this study, they looked at development over time. So it looked like the viruses also were delayed, but it looked like that the delay was mostly because the bacteria were delayed in development.
So it's dependent on the bacteria.
Yes.
Okay, thank you. Are there any other questions, Ray?
I'd like to ask one question. Last one is that you have mentioned that 1 ml of seawater contains 10 to the power 7 viruses. And we often go to the sea beach and swim in the sea water. So is there any chance of having viral infection from the sea water?
Well, a lot of the viruses in the seawater are, a lot of bacteria. How do they
survive in the increasing salinity in the seawater?
Yeah, there are a lot of phages and a lot of, really a lot of all of the different type of viruses, but the majority are phages. But humans swim in the ocean all the time, and so I think the risk of being infected there would be just like the risk of being in the environment, and probably the viruses that infect us most we get from other humans. So contact with other humans will give us those viruses.
Are there any other questions? If not, thank you very much. We have a little extra time for lunch. We'll reconvene 1:30. I think it's time to begin again. It's a real pleasure for me to introduce my very good friend, Joseph New, who's a professor of pediatrics at the University of Florida and a physician scientist in neonatology. He's had a lot of effort in gut microbiome, and today he's going to talk about gut microbiota, host gene involvement, and cell trafficking in the milk. Joe.
Thank you, Alan, and I want to thank both Alan and the Nestle Nutrition Institute for inviting me. This is a real honor, and I think it's been an excellent meeting so far. I've learned a tremendous amount. I do want to say that this lecture is not going to involve as much about gene trafficking as things that I'm actually a lot more familiar with. So I'm just going to get started here with the agenda. I'll briefly talk about human milk, fresh versus donor human milk composition. This is something that is actually quite an area of controversy right now. Talk about some of the microbiota in human milk and its implications, microbial metabolomics in the developing intestine, microbial effects on the innate and adaptive immune systems, and multi-omic approaches to evaluate microbe-host interactions. So anybody know who this guy is? Yeah, Feynman, Richard Feynman. He's a quantum physicist from Caltech. He won the Nobel Prize. And I just want to give a couple of his quotes. He has a lot of very interesting quotes, but here is one. Religion is a culture of faith. Science is a culture of doubt. We must be careful not to believe things simply because we want them to be true. No one can fool you as easily as you can fool yourself. And I think that this is very true for many areas that we've been discussing, especially when it comes to human milk. So I'm going to start with a quiz. And I know many of you are basic scientists, and there are a few neonatologists here. And I'm just going to ask you for this 25-week gestation, 2-day-old preterm infant, which of the following is or are false? Enteral feeds should be initiated. This is at 2 days. Donor milk should always be the first feeding because the mother is often only able to produce small amounts. Bank donor milk composition is similar to that of the baby's own mother's milk. And it has clearly been shown in head-to-head randomized trials that banked donor milk provides similar benefits to baby's own mother's milk. Okay, so 2, 3, and 4 are false. Number 1 is somewhat controversial, but I think that over the last few years, most neonatologists are beginning to enterally feed low birth weight babies. But what should we be feeding them? In 2012, in the US, the American Academy of Pediatrics came out with these guidelines. The potent benefits of human milk are such that all preterm infants should receive human milk. Mother's own milk, fresh, frozen, should be the primary diet, and it should be fortified appropriately for the infant born weighing less than 1.5 kilograms. If mother's own milk is unavailable despite significant lactation support, pasteurized donor milk should be used. Quality control of pasteurized donor milk is important and should be monitored. Okay, so this is what the American Academy of Pediatrics told us neonatologists, and we all started, most neonatal intensive care units took this very seriously, and either donor milk or the baby's own mother's milk was being used and formula was not being used. So over the next few minutes, I want to address a few questions. First, can these guidelines potentially lead to harm? Is there a rationale to use mother's own milk versus donor milk? What components are lost with routine pasteurization of bank donor milk, and what is their importance? And how can we make donor milk more like mother's own milk? So let's start with potential harm. This is some work that was done at the University of Florida in our neonatal intensive care unit. And we looked at some data prior to the 2012 guidelines, and then after the 2012 guidelines. And here, what we are seeing in this first column is prior to donor milk implementation, and this is at week 1, 2, 3, 4, 5, and 6 after birth. And here we are looking at the decrease in the percent of own mother's milk use. So what we are seeing over here in week 1 is a decrease, 2, a decrease, 3, a decrease, all the way down. We're seeing a decrease in the use of the baby's own mother's milk, and donor milk was being used much more prevalently in these babies. Why? Well, it became much more convenient to use bank donor milk. And it was available, and the mothers started to believe that, oh, this is the same thing. The nursing staff believed this is the same thing as the baby's own mother's milk. And so we started to start questioning this whole practice a little bit. Now, what about donor milk? In the United States, donor milk is pasteurized, 62.5 degrees centigrade for 1/2 hour. And that actually gets rid of some very important components. This makes us realize that, at least to some extent, as far as the composition of donor milk and baby's own mother's milk is concerned, that they are very different from one another. This is given to me as courtesy from Dr. Peter Hartman from University of Western Australia, and here we see some of the compositional factors in donor milk. And here we see some of the things that are highly abolished. Here is alkaline phosphatase, bile salt stimulated lipase, almost totally abolished with the pasteurization process. IgA, markedly decreased. So what are the implications of this? Bile salt stimulated lipase, considered to be a very important enzyme in terms of hydrolysis of triglycerides. And if you don't have this available, you do not have the most efficient hydrolysis of the triglycerides. So that's an important nutritional factor. What about the alkaline phosphatase? Breast milk alkaline phosphatase decreases over time. So here on the x-axis, we have days post-birth, and here's alkaline phosphatase activity, initially quite high and then decreasing. Well, what does alkaline phosphatase do? There's an intestinal alkaline phosphatase, and there's a human milk intestinal phosphatase. Intestinal phosphatase has the capability to dephosphorylate various pro-inflammatory microbial components, including lipopolysaccharide, thus making them unrecognizable by toll-like receptors. And as we go on in this lecture, you will recognize that toll-like receptors are very important. Alkaline phosphatase can directly inhibit the NF-kappa B pathway components and thereby downregulating inflammatory responses. There are some studies that have been done in animals, in mice, looking at the effects of recombinant alkaline phosphatase on experimental sepsis. So here, in this study, they looked at 2 outcomes. Here's the body temperature, and here's percent survival. What happens during sepsis in this animal model, if you induce sepsis by giving LPS, these animals start getting cold, like they're beginning to die. And here what we're seeing is that without the alkaline phosphatase, these animals start getting quite cold. But the alkaline phosphatase appears to rescue it. Here is the survival without the alkaline phosphatase, with the alkaline phosphatase. So this is considered to be an important component in the human milk. Cells, live cells, are killed in the pasteurization process. And I'm really not going to get into the cells very much because others have already gotten into that, and I know that there will be more discussions about this in the future in this conference. But I do want to talk more about the microbes in human milk. And Sam really did a very nice job this morning about talking about what is known about microbes in human milk, and I'll just talk about it a little bit more here. In the past, prior to about 10 years ago, when we had a milk sample, or if we had a mother who we were concerned about that the milk might not be right, we cultured it. And if we found microbes in that milk, we would throw it away. Okay? Now we're beginning to recognize that perhaps that these microbes might actually play a role. So studies using both culture-based and non-culture-based techniques show that there are endogenous microbes in human milk and that they could be beneficial, as we discussed this morning. Here's the classic study from Hunt and colleagues. And what they looked at in this study, and I'll go into this in a little bit of detail here, is really interesting. Each one of these S's represent a different mother. So there are 16 mothers in this study who are expressing their milk at 1, 2, and 4 weeks after birth. And each one of these colors represents different taxa of bacteria. So in this particular mother, S1, you can see that the colors are quite similar. They change a little bit in terms of their quality and also amount, but this mother, over time, these microbes are about the same, but they're very different than this mother. They're very different than this mother. So there appears to be a personalization of these microbes in these different mothers. This is work that was done in Spain, looking at the quantity of microbes, the total quantity of microbes in milk. And this is the mean plus or minus standard deviations, and these are the individual mother's concentrations of milk. So here is the colostral, transitional, and mature milk, and you can see some of these mothers had high quantities of microbes, up to 10 to the 8th microbes per milliliter in their milk. This is the mean quantity. So there's a lot of microbes in human milk. Now, I want to digress a little bit and talk about probiotics, and this is something that was brought up this morning, and neonatologists don't like to talk about this because it's sort of like talking about the president in the United States. If you talk to the wrong people, you can get into big trouble, okay? But what has happened, this is Sisyphus going up the hill, pushing the ball up the hill, this big boulder up the hill. And in 2010, we seemed to have a tipping point for probiotics. A meta-analysis came out in the journal Pediatrics, and they had 11 different probiotics being used in a meta-analysis. And this meta-analysis with these 10, 11 different probiotics suggested a decrease in necrotizing enterocolitis and also death. And at the end of the discussion section in this meta-analysis paper, they said something that you almost never hear of in a meta-analysis. They didn't say more studies are needed. They said, it's over. We need to start probiotics in all of our babies. Okay, so this was 11 different probiotic species that were being used. So then the question arises, are we really there yet? Well, several countries started to use this routinely, and different probiotics were being used. In fact, in the United States, the most commonly used probiotic is something called Lactobacillus rhamnosus, or Culturelle. 15% of the neonatal intensive care units are using probiotics routinely to prevent necrotizing enterocolitis. But Culturelle was never even one of the probiotics that was studied in that meta-analysis. And so the point is that there are different probiotic preparations, and not one of them, until fairly recently, has been studied in terms of prevention of necrotizing enterocolitis. The one study that was done in the UK evaluated around 1,400 babies and found no decrease in sepsis, no decrease in necrotizing enterocolitis, or any other adverse outcomes. So we have one randomized controlled study. I was asked by the Journal of Pediatrics to discuss, shortly after the meta-analysis came out, the routine probiotic usage, and I wrote this commentary, Routine Probiotics for Premature Infants. Let's be careful. Several reasons that we need to be careful. Number one, meta-analyses. Many meta-analyses, after a couple of years, are proven to be false. Okay, the results are proven to be false. They are, especially if they are based on observational studies or really small, non-powered randomized trials. Pooling of mixed data across trials as if they belong to a single large trial can be a problem, especially when you are using different preparations and if you are saying that they're all the same, which they are not. Are probiotics a food supplement or a drug? Well, it depends. If you are using them to prevent a disease, such as in premature infants, if you are using them to prevent a disease such as necrotizing enterocolitis, this is a medical claim. And this means that they should undergo rigorous testing. I know that the FDA and the European agencies do require rigorous testing if you are going to prevent a disease. We also have had some problems, and this is just one of the problems. A couple of years ago at Yale University, they started to use probiotics because of a high incidence of necrotizing enterocolitis, and they saw that there was one baby who ended up dying, and that baby went to pathology. They looked very closely, and the pathologist found that the baby had mucormycosis, and they traced that back to the probiotic preparation. And so this is something that is quite scary. If you don't have correct standards for a certain preparation, pharmaceutical standard for preparation. Now, let's get back to the human milk. I showed you the picture before, the illustration that human milk has high quantities of microbes that may be personalized. If you assume an intake of 800 milliliters per day, that's in a term baby, and you assume the average number of bacterial cells, which is 10 to the 5th to the 6th, this will provide about 10 to the 7th to the 8th bacterial cells daily, which is close to the dose used in most probiotic studies. So you are actually providing a mixture of microbial agents in the human milk. So what about these? Could they potentially be truly personalized? Well, if you have a baby who is close to the mother, and this is work that was done by Dr. Walker and Kleiman, published in 1979, on this system that we have heard about before several times, the enteromammary system. And if the babies are close to the mother, and if the mother picks up some of the microbes that the baby might be colonized by, the baby's immune system may not be able to completely respond, but the mother's immune system is. And these immunoglobulins, cells, etc., will then go into the breast milk and get into the baby's gastrointestinal tract and provide some potential benefits. So this is the enteromammary system, and we've been discussing this in terms of both immunology and the types of microbes that might be available. So in the late 1970s, when this review was done, a lot of this work came from studies that was done in animals. But I think we're beginning to recognize there are at least 3 studies now suggesting that this is actually something that does happen in humans. This is a study that was done by Riskin and colleagues in Israel, and they looked at 12 infants with viral disease versus controls. They looked at the milk samples longitudinally, taken 7 days apart, while the infants were recovering from their illness. And so they analyzed the milk samples for CD45 leukocytes, macrophage counts, TNF-alpha. They found that all of those decreased. The lactoferrin also was changed, but it was a borderline effect. So they concluded from this study that this supported a dynamic nature of immune defense in breastfeeding infants. So this closeness to the mother, sort of like in kangaroos, closeness to the mother, kangaroo care or skin-to-skin care may actually be very important in terms of this dynamic interplay between the mother and the baby. Here's a preterm baby, the kind that we take care of a lot of in the neonatal intensive care unit, and At the bottom of the slide, you see a baby with a very distended abdomen. This is a baby who has developed necrotizing enterocolitis, one of the most dreaded diseases that we see in neonatal intensive care. This kills about 30% of the babies that develop it. But we've known about this disease now for over 50 years, but we have not had that much progress in the disease. Why? Well, here are some of the reasons. We really don't have a good definition of this disease yet. We lump several different entities into one disease that we call necrotizing enterocolitis. But I think we're beginning to recognize more and more that necrotizing enterocolitis is like diabetes. There's type 1 diabetes, there's type 2 diabetes, and there's diabetes that are in between. So what we are calling necrotizing enterocolitis is more than one disease. And we need to delineate and better define the disease before we can move forward. We have animal models that were developed in the late 1970s, and there are over 200 publications that you can find in PubMed using some variant of this animal model, which does not at all represent what we see in neonatal intensive care. And we've also focused on individual pathways, like the TLR4 pathway, for example, which is probably very important, but we can't just focus on one pathway. We have to focus on an entire system rather than on one pathway. So this is where we are going in the future. This picture is from a publication that Dr. Walker and I wrote in just a few years back. And at the top, you can see a baby with necrotizing enterocolitis, distended abdomen, some erythema around the belly button, and the radiologic findings. This is what we see in terms of air in the bowel wall, something called pneumatosis intestinalis, and sometimes we will actually see air in the liver, the portal tract of the liver, and this baby ended up having a piece of dead bowel that was taken out. Unfortunately, this baby did okay after this resection. So we need to think about this in terms of several different diseases, but I would like to sort of focus on more of the inflammatory disease that we see in preterm babies. And with this inflammatory disease that we see in preterm babies, if we look at gestational age of the baby's birth, we find a difference in terms of the time that the necrotizing enterocolitis develops. So here we have a baby born at 23 weeks gestation. Here we have a baby born at 29 weeks gestation. And the question is, when will this baby develop necrotizing enterocolitis most likely? And when will this baby develop it most likely? Well, this particular graph has been repeated by several different groups, National Institutes of Health, the private network in the United States called Pediatrics with an X. This is a study that I did with Dr. Mohan Pami from Baylor University. And what we are seeing here is that the corrected gestational age in weeks is highest between about 28 to 32 weeks gestation. So what that means is that in all likelihood with this classic form of necrotizing enterocolitis, this baby would develop it at about 6 weeks of age, and this baby would develop it at about 2 weeks after birth. We don't know why this happens, why this peak seems to occur, but there are several speculations. One speculation has to do with the microvasculature of the gastrointestinal tract. Similar to the eye, the microvasculature of the gastrointestinal tract might be developing in a very similar route. We rarely see retinopathy of prematurity develop prior to 30 weeks gestation. That is because of the development of the blood vessels in the eye. The same thing may hold true for the gastrointestinal tract. Dr. Walker and Dr. Hackham and different colleagues have shown that there is a toll-like receptor 4 that is present in the gastrointestinal tract that responds to lipopolysaccharide. That's what I showed you before, responding to the alkaline phosphatase. Well, that lipopolysaccharide seems to be at its, the toll-like receptor seems to be at its highest concentration in the last part of the second trimester of pregnancy. Keep that in mind as I continue to talk here, because this is going to be important. What about the microbes that get into the gastrointestinal tract? How do they colonize the gastrointestinal tract? Well, this is a combination of studies. This is actually a meta-analysis done on sequences from 10 different studies where the gastrointestinal tract of premature babies, the feces was looked at in terms of 16S sequencing. Dr. Pammy and I did this study. Initially, I didn't think that this could be done, but we took sequences from the different neonatal intensive care units from Boston, from University of Chicago, from Cincinnati, from various places where they had done these studies in preterm babies, and then we looked at the taxonomy over time. Here are the control babies, here are the necrotizing enterocolitis babies, and here we see in the controls that this blue and this yellowish color here are somewhat similar over time. But over time, in the babies who develop necrotizing enterocolitis, the blue increases and the yellow seems to decrease. What are the blue? The blue are a phylum of bacteria called the proteobacteria. The yellow are a phylum of bacteria called the firmicutes. We also see over here in the controls, this yellow, the bright yellow at the bottom. These are the Bacteroidetes. So we see some Bacteroidetes over here, but very few Bacteroidetes over here. So we can speculate on this. We don't have any causality, but we can do some speculation. So let's speculate a little bit about the proteobacteria. These are the bacteria that appear to increase prior to the development of necrotizing enterocolitis. These are gram-negative bacteria that are high in lipopolysaccharide in their cell walls. And the abundance of proteobacteria increased prior to the development of necrotizing enterocolitis. Representatives of this phylum are Klebsiella, Pseudomonas, E. coli, high levels of lipopolysaccharide in their cell wall. Here's another phylum, the firmicutes. Firmicutes actually decrease prior to the development of necrotizing enterocolitis. And the firmicutes are largely gram-positives, and lactobacilli are a common class of the firmicutes phylum. These are high in lipoteichoic acid. They also have a high capability to produce short-chain fatty acids, such as butyrate, propionate, and acetate. Butyrate is thought to be very important in terms of a fuel for the colonocyte. Butyrate also tightens junctions, interepithelial junctions between cells. Propionate is thought to be very important in terms of the FOXP3 system and production of anti-inflammatory mediators. And then the Bacteroidetes, also thought to be very important. And remember, in the neck babies, the babies who develop necrotizing enterocolitis, very few Bacteroidetes. Sarkis Kazmanian has shown this some work that he did looking at the Bacteroidetes. There's a certain immunomodulatory molecule called polysaccharide A in the cell wall of these microbes. And these are very important in the conversion of CD4 cells into the FOXP3 cells, and these regulate the production of IL-10. So these can be immunomodulatory. And these are the types of microbes that were not present in those babies that developed necrotizing enterocolitis. So we have kind of an interesting story developing here. It's not proving any kind of causality, but it's an interesting observation and association. So what do we do in the neonatal intensive care unit that might actually cause necrotizing enterocolitis? And here we have a pogo cartoon that says we have met the enemy and the enemy is us. And the type of diet may be important. Human milk, baby's own mother's milk versus donor milk versus formula. That may be very important. What about antibiotic usage? That may be important too. Or even the lack of enteral feeding. Not putting any fuel into the gastrointestinal tract might be important. Well, what are the most commonly used antibiotics in neonatal intensive care? What are the most commonly used drugs in neonatal intensive care? Here we see 10 of them. Of these, at the very top, by far, gentamicin and ampicillin are the most commonly used drugs in the United States. But then just a little bit further down, you see cefotaxime, Vancomycin, also very commonly used drugs. Can this have an effect on the gastrointestinal tract? There are at least 3 studies now suggesting that the number of days on antibiotics increases the relative risk for the development of various adverse outcomes, including necrotizing enterocolitis. So, according to this study, the average length of treatment, which is about 7 days, increases the risk of necrotizing enterocolitis by about 50%. This is a quote from a colleague of mine, a neonatologist. So I give a couple days antibiotics to my preterm patients. What does that matter if I change the microbes in the GI tract, since I could potentially be saving the baby's life by treating unrecognized early onset sepsis? Let me just start by saying babies less than 33 weeks gestation, prior to a couple of years ago, prior to some of these practices, quality improvement practices that we've been starting, almost 100% of our very low birth weight babies were getting antibiotics. It's slowly beginning to change, but they get at least 2 days of antibiotics. So what does this do when they get at least 2 days of antibiotics? Well, here what we're seeing is treated babies, and here are controls. These are different taxa of bacteria, and we see that in the controls, here's the proteobacteria, and I'm sorry, here's the firmicutes in the controls, and in the treated, I'm having a hard time reading this. Yeah, there's a marked difference. Here's the proteobacteria in the controls, much larger proteobacteria in the treated animals than in the controls. You find a similar relationship at week 8. So 2 days of antibiotics shortly after birth will alter the microbiota at 4 weeks and at 8 weeks. At Washington University, Barbara Warner and colleagues have shown that even up to the first year, you have antimicrobial resistant microbes, more so in those babies who are treated with antibiotics than you do in the non-treated babies. 2 months ago, this paper came out of, it was published in Nature Medicine, and this was a very interesting study that looked at IgA, and we were talking this morning about microbes that were coupled to antibodies. This study evaluated microbes coupled to IgA. And they did flow cytometry and looked at the stools of babies who were being formula fed versus getting their own mother's milk. And here what we see is that more IgA in those babies who were getting their own mother's milk compared to formula. Okay, here we're looking at the intestinal bacteria bound, total intestinal bacteria bound by IgA. Okay, and this is time after delivery on the x-axis. Here we see the formula-fed babies. Here we see the maternal milk-fed babies. And here we see this increase where you didn't see this increase over here. It's pretty much the same because these babies were getting the IgA in their mother's milk. This is probably the intestinal IgA that's being produced by the baby's own intestine. Here we see necrotizing enterocolitis, controls versus the necrotizing enterocolitis babies. And we see much lower IgA-bound microbes in the NEC babies. Here we see the types of bacteria and necrotizing enterocolitis babies versus the controls. And here they looked at the bacteria at a family level. Before I showed you the bacteria at a phylum level, here we see the Enterobacteriaceae, which is at the family level, much higher in the necrotizing enterocolitis babies. Well, what about specific Enterobacteriaceae bound to IgA? Here we see the intestinal bacteria in the controls, and here's the NEC babies. These are about the same, and here they decrease over time. Here's the lower binding in the necrotizing enterocolitis babies. So some very interesting studies that suggest differences in IgA. I know that we're going to hear more about this tomorrow afternoon. Now, what about C-section versus vaginal delivery? This is something that was brought up several times in this conference. Now, it's thought that babies who are born by vaginal delivery will pick up the vaginal microbes in their mouth, and these will get into the baby's gastrointestinal tract and then colonize the gastrointestinal tract. And those babies who are born by C-section delivery do not undergo this process. There are several studies that have been done that seem to support this view. Here's probably one of the more classic studies done by Dr. Dominguez-Bello from NYU. This was a study that was done in Venezuela. And it was done on actually very few subjects, but you can see clustering of C-section delivered babies and mother's skin over here. And here we see clustering of the vaginally delivered baby's stools and the vaginal microbes, okay, suggesting that those babies who went through the vagina had vaginal microbes in the stool. So this led to something very interesting. This is a paper that was published in Nature Medicine a little bit over a year ago, and the title of this was Partial Restoration of the Microbiota of Cesarean-Born Infants via Vaginal Microbial Transfer. So what do you do with this? Well, if you have a mother who delivers by cesarean section, you put a swab into that mother's vagina, and then you take that swab after leaving it there for about an hour, and you swab the baby's mouth and the face and the chest. And they found that they could actually get the vaginal microbes into the newborn's gastrointestinal tract by doing this. There are many parents that are beginning to ask obstetricians to do this, okay? But we've been a little bit afraid to begin to initiate this. This is the American College of OBGYN in 2016, and I just want to read the red part to you. We recommend against the practice of vaginal seeding until better data are available establishing the safety and benefits of this practice. So just imagine if a mother had unrecognized herpes virus infection, and if you inoculated that baby with a herpes virus infection, that would be primary herpes, and the baby could potentially die from that. Certainly that's not something that we want to do to our babies. Last week, this article came out in Nature. Stunted microbiota and opportunistic pathogen colonization in cesarean section birth. Okay, have any of you seen this paper yet? That's a fascinating paper. Here is some of the data from that paper. Delivery mode seems to make a big difference in terms of the microbiota development. But the question that was brought up by Sam Ratava this morning was, what about antibiotic use? Well, almost all mothers who deliver by cesarean section get antibiotics around the time of C-section delivery. And that did have somewhat of an effect. Postnatal stay in the hospital seemed to have an effect. Breastfeeding seemed to have an effect. Hospital site. There are many different factors that were associated with C-section delivery, not necessarily the C-section itself. Of interest was the fact that in this study, the bacteria that they saw in the vaginally delivered babies did not consist of a large quantity of the lactobacilli that you see in the vaginal tract. Saw other kinds of bacteria. Here are some of the bacteria that they saw, and here we see in the vaginally delivered babies, you can see this pattern, and in the C-section delivered babies, you see this pattern. So there are some real differences early on from 4 to 7 days, but then 21 days, they begin to even out, and then in infancy, they look very much the same. But even at 8 months, the Bacteroidetes appear to be significantly more present in the vaginally delivered babies compared to the C-section delivered babies. I still think that there's a lot of confounding factors in this study, and we have to think about the stay in the hospital. A baby who is delivered by cesarean section not only gets the antibiotics compared to the vaginally delivered baby, but that baby stays in the hospital for several days longer than the baby who is delivered by vaginal delivery. Okay? The types of microbes that they were seeing in the C-section delivered babies were hospital microbes. Klebsiella, E. coli, etc. So that's certainly something to take into consideration. Slow initiation of breastfeeding also may be very important. Those babies who are born by cesarean section, the mothers do not provide the same amount of breast milk early on as do the babies who are vaginally delivered. So it may take a little longer for that seeding to occur and for the types of microbes and the other components of breast milk that may actually promote the microbiota in the human milk to develop. This is a study by Megan Azad from Canada, and I just want to show you here, this is a very small number of subjects. She has actually done more work recently, but these are vaginally delivered babies, cesarean section delivered babies who are not breastfed, and at 4 months you see a difference in the microbiota. But those who are breastfed, you see very little difference in the microbiota, suggesting that breastfeeding seems to reverse this effect of C-section or stay in the hospital or whatever it is in those C-section delivered babies that are causing differences in the microbiota. So in the future, some questions. Can the microbes in human milk overcome antibiotic-induced or other forms of dysbiosis that are associated with necrotizing enterocolitis? Can the microbes in human milk overcome the hospital environment effects associated with C-section deliveries? Should we be focusing on the use of fresh mother's milk rather than bank donor milk? And can we improve our scientific approaches? Let me just get into a couple of these areas here. Work that we did at the University of Florida just a couple of years ago involved the question of donor milk versus baby's own mother's milk. We know that there's a very rich microbial milieu in baby's own mother's milk, but donor milk is pasteurized. A lot of mothers cannot produce a lot of milk right after birth. And so is it possible that we might be able to take some of that small quantities of the baby's own mother's milk and put it into banked donor milk and develop a composition of microbes that is very similar to that of the baby's own mother's milk? So this was called a transfaunation process, and we evaluated the quantity of microbes in baby's own mother's milk using both culture-based and non-culture-based techniques. And we also used non-culture-based and culture-based techniques on the donor milk, found almost no microbes in the donor milk in terms of live microbes, but we were able to find microbial DNA in the donor milk. So here is donor milk versus baby's own mother's milk, and you can see that baby's own mother's milk has more microbes, different types of microbes. I know you can't see that slide very well, but in this study, the major result was incubation of donor milk with 10% mother's own milk for 4 to 8 hours resulted in levels of microbes very similar to that of mother's own milk, done by both culture and non-culture-based techniques. So that's one way we could potentially improve the microbial environment, but we have to also show that it matters in the future. So we might be doing something that just doesn't matter at all, but we have to show that in the future. What about improved scientific approaches? Well, when we do microbiome studies, when we do metabolomic studies, all of these show associations. This is a problem. We can look at our genome, we can look at our microbiome, we can look at the metabolites, we can look at the epigenome, we can look at them singly, but this is only association type of work. So here is interesting story. This is an old Hindi, I think it's Hindi, parable. And good presentation should be susceptible to only one interpretation. I'll read this to you. It was 6 men of Indostan, to learning much inclined, who went to see the elephant, though all of them were blind. To each by observation might satisfy his mind. The first felt the side, and he said, "This is like a wall." The second felt a tusk, and said, "This is very like a spear." The third felt a trunk. "This is very like a snake." The fourth felt a knee. "This is very like a tree." The fifth felt an ear. "It is mighty like a fan." The sixth, it felt like a rope. And so the men of Indostan disputed loud and long, each in his own opinion, exceeding stiff and strong, though each was partially in the right and all were in the wrong. So this is an area that I think we need to just not just look at one component like the microbiome. We need to look at multi-omics. And we are now getting the bioinformatic technologies to help us look at multi-omics. So why do this? Each individual type of omic data may provide a list of differences associated with the disease. Individually, they can be useful, but analysis of only one data type is limited to correlations, mostly reflecting reactive processes, chicken or egg first argument, rather than causative ones. These can be hypothesis generating, but not hypothesis testing. These are fishing expeditions, as one would hear in a study section at National Institutes of Health. Integration of different omics data types more strongly identify patterns associated with that are associated with and perhaps are even causative of the disease. And this can more readily lead to disease treatment targets. So I'm going to just spend a few minutes here on antibiotic-induced dysbiosis. Remember I said before that we have some problems with giving a lot of antibiotics to our premature babies and also to mothers, as Sam said this morning. And this can induce something called a dysbiosis. What are we really doing with these antibiotic treatments? At the University of Florida, we had the fortune of having a small National Institutes of Health grant to do one of the first randomized controlled studies of antibiotics in preterm babies. And it was a study that was called a pragmatic randomized trial. There are certain babies that neonatologists would never agree not to give antibiotics to. Okay, this was the high-risk group over here. So if the mother had definite chorioamnionitis, a fever of 39 degrees centigrade, we would have to give antibiotics to those babies. But here we have this group here, the randomized group, Group C, where one group, they were either randomized to get antibiotics or to not get antibiotics. And I just want to show you a little glimpse of the data that we're getting. And this is, we're attempting to get multi-omics data on these babies. So here at the top, these are somewhat complex diagrams, but here at the top, we see the antibiotics that were used. This is a baby who got antibiotics, who was randomized to get antibiotics early on in the course. Okay, so you see different antibiotics uses over time, over this baby's hospital course. These circles here represent the microbes, the stool microbes, and the different colors are different taxa of microbes. The first stool, meconium, you can see very low diversity. The size here is done on an exponential scale, and that is the total number of microbes, the total microbial quantity. And you can see the total microbial quantity here increases. We see some changes with the use of antibiotics. So here's an antibiotic usage, and here we see this shift in the types of microbes. Here we're looking at different inflammatory mediators, and down here we are looking at the metabolomics. Here I'm just looking at the butyric acid, but you can see butyric acid changing over here depending on the use of either the type of milk that the baby is getting or the antibiotics. So this is very complex, and here is another baby that did not get antibiotics right offhand. Again, meconium, fairly high quantity of microbes, but very low diversity. That begins to change over time. Here the baby got some antibiotics because the neonatologist thought the baby was getting sick. You can see a major shift in the types of microbes. Here the baby goes from donor breast milk to formula, and we're seeing different types of microbes over here. Here's the inflammatory mediators in this stool. Here's the butyrate in this stool. Just a different way to look at this from a more multi-omic perspective. And here's metabolomics and clustering. And here what we're seeing is, here is the randomized group that is getting antibiotics. This is the group that is definitely getting antibiotics in the middle here. These are the babies whose mothers have chorioamnionitis. And here's the group that was randomized to getting antibiotics and no getting antibiotics. And you can see that these three groups here are very different from one another. So the use of antibiotics changes the metabolome in the gastrointestinal tract of these babies. In the future, we also want to look at transcriptomics. How do we look at transcriptomics in the human? Well, you can't do biopsies on these babies, but we have some colleagues at Texas A&M that are able to get sloughed epithelial cells, and we're able to look at microbial transcriptomics in these sloughed epithelial cells. So in the future, we hope to add transcriptomics to these studies. So let's put it all together, and here's some considerations for design of multi-omic studies. These are best aimed at complex processes or diseases, not those where known single genes or bacteria cause disease. These are really going to be a challenge to do these types of studies, but I think that they are possible. Complex diseases develop over time. Several time points need to be considered, especially when you're talking about babies. Biobanks are needed because usually large number of samples are needed. Sample size determinations are needed before collection of data. So I'm going to summarize here. There's numerous bioactive factors, including live microbes and microbial components in human milk, but their actual function remain poorly understood. They're based on associations rather than causal mechanisms, so we have a lot of circumstantial versus direct evidence. Studies using multi-omics in the future will better provide causal mechanistic evidence in human studies, where we may not need to go to germ-free animals in some of these studies that have been done in cell cultures, where we have to take a leap of faith. I'd like to acknowledge many people who I've had the opportunity to work with over the years and funding sources, and thanks to the people I work with in the neonatal intensive care unit at the University of Florida. And thank you for your attention.
We have a little bit of time for questions.
Thank you for this wonderful talk. Vanessa from UC Davis. I'm wondering if you could comment on the idea of potentially better matching lactation age with donor milk for the gestational age for the babies that are receiving it.
I think that's a really good idea, and I know that when I visited Spain a couple of years ago, there was an attempt to do this, where they were actually trying to match the lactational age with the particular baby. So I think that's a great idea. The problem is that the milk is still pasteurized, and we don't have the same factors in the donor milk as we have in baby's own mother's milk. Yes.
Thank you for wonderful lecture. Are there some biomarkers of NEC, and were there some efforts to use fecal microbiota transfer for at the beginning of the disease?
So to your second question, fecal transplants for trying to prevent necrotizing enterocolitis, no, that has not been done. It's been discussed, and I think that there's a lot of discussion now about finding consortia of microbes, not actual fecal microbial transplants, but actually finding the right consortia of microbes that may be working together to provide a beneficial effect, not just one or two or three probiotic agents. Your first question, the biomarkers for the disease, there are some biomarkers, but I think until we get a better idea of what the definition of the disease actually is and separate out the different subcategories of this disease, are we going to be able to use the biomarkers to any reasonable extent?
Thank you. Thank you very much for your nice deliberation. I'd like to ask, what is the policy in your unit regarding the babies of very low birth weight and extreme low birth weight who does not have any risk factor in the mother during delivery or before delivery? Do you use, prophylactic antibiotic, or you do not use in your intensive care?
So, in premature babies,
very low birth weight and extreme
very
low
low
birth
birth weight babies. If you would have asked me that question 5 years ago, I would have said they all get antibiotics. Now, in 2019, we are having some babies that we are getting very selective about, and we are beginning to be a little bit more cautious. So if a mother does not have risk factors, if the baby is born, preterm, 27, 28 weeks gestation, and looks fairly good, even if the baby has to go on mechanical ventilation, we are watching those babies very closely and not necessarily treating all of them with antibiotics.
But in our unit, we have seen that baby is looking very good in first 1 or 2 or 3 days, and they didn't develop respiratory distress syndrome. Everything is okay, fine. And on the 3rd day, 4th day, the baby is getting reluctant to feed and abdominal distension and signs of sepsis. And in that case, then we have to start again antibiotic and your immunoglobulins or all this we have to start. So still it's a and you have analyzed the microbiota in these stools when the babies are receiving antibiotic or not receiving antibiotic. But in terms of survival, do you have any data? In
terms of what?
Survival of the baby
Survival, yeah.
using antibiotic, prophylactic antibiotic, or not using prophylactic antibiotic?
No, we don't have that data in terms of survival with the prophylactic antibiotic usage. We're just beginning to ask that question right now. But in terms of, just to give you an example, the study that we did on necrotizing enterocolitis and looking at the microbiota at the University of Florida, we had 3 different hospitals. We obtained samples from 900 babies, and we did blood cultures on all of those 900 babies. 1% of them had positive blood cultures, and several of those were probably contaminants. So in other words, the blood cultures don't help us very much. This is a real conundrum. And so should we even be doing these at the beginning? So I do understand what you're saying. Occasionally you will have a baby who ends up developing signs of sepsis or getting sicker or may even become septic. But are you going to treat all of those babies? We had one baby who was on our study who we actually was randomized to not get antibiotics, but the baby started looking pretty bad within about 4 hours, and we bailed that baby out. We did have the opportunity to bail the baby out. That baby started on antibiotics. We gave the baby ampicillin and gentamicin. The baby died. But the baby died of a bug that was resistant to ampicillin and gentamicin. So even though we gave the baby antibiotics, the baby had a resistant Klebsiella.
There might have other causes for there might be some other causes, metabolic disease, all this, which behave like a septicemia, but there may be other cause of death.
Yeah. Nick?
Thanks, Joe. Another great talk. Nick Hamilton from Newcastle. Can I just ask you about oral antibiotics? You remember there were a series of studies about 20 years ago showing reduction in neck. And as part of a large collaboration, we just published data showing that the babies who don't get antibiotics, preterm babies who don't get antibiotics, actually have a higher risk of neck. So given that neck seems to be associated with this sort of bloom of proteobacteria, do you think there might still be a role for damping down the proteobacteria with potentially orally administered gentamicin or something like this for a few days?
The studies that were done, this was 30 years ago, You're not that old. So these studies were done around 30 years ago, and there were, I think, 3 or 4 of them that looked at oral kanamycin and oral gentamicin. And they were really small studies, very underpowered studies. And one of them was done at the University of Florida. I was not involved with that, but it was done at the University of Florida. And one of the things that occurred, they started giving oral kanamycin routinely to all the babies in the neonatal intensive care unit. They started to see resistant microbes. And the resistant microbes did not only occur, that they started seeing it not only in the neonatal intensive care unit, but other parts of the hospital as well. So that caused us to have a lot of reservations about continuing with that.
But in the piglet models that Per Sangilde has done, the most effective intervention for preventing NEC currently is oral antibiotics, better than probiotics, better than everything else.
Let me say, it was brought up earlier in this conference that certain animals have a very different immune system than the human. And I remember working on piglets many years ago when we were looking at cholesterol in the microvillus membrane of these animals, and we would deliver them by cesarean section, take them into a clean environment, and within 1 week they would be dead. And we would have to either let those animals have one milk feeding or give them immunoglobulin. They did not get that transfer through the placenta as we humans do. So I think that this is a different immune system, and I have a hard time really, I shouldn't say believing. I think that they're well-done studies. It's just that the whole premise of the study it's very different than the human.
Go ahead.
Do you know, well, you mentioned the LGG for prevention of necrotizing enterocolitis. I'm aware of one study which showed that there was absolutely no effect, and on the contrary, there was an increase in sepsis or infections. But Deshpande in Sydney, Australia, they used 2 probiotics. Do you know, and apparently they're using them routinely now, do you know what is the current status of prevention of NEC out of studies in these centers?
Well, there are, in terms of the usage of the probiotics, in the U.S., as I mentioned, about 15% of neonatal intensive care units have been using probiotics. The most commonly used probiotics is the Lactobacillus rhamnosus. The studies that have been, like you said, have not shown a decrease in necrotizing enterocolitis. One study that was done at Emory by Ravi Patel, this was a study where they looked at giving no probiotics and then starting with the Lactobacillus rhamnosus, 2 different time epochs. The incidence of necrotizing enterocolitis actually increased after starting the Lactobacillus rhamnosus. With 2 different probiotics, Infloran, in Europe, there was one study that showed almost the same thing as the Emory study, that if you look at the lack of probiotic administration versus after probiotic administration, there was an increase. But that's not been seen uniformly in all studies. So it's just something that I think we need to take into account.
I think we're going to stop now and take a coffee break. If you have additional questions, you can ask Joe. Okay, thank you.
talk. We are about to start. And we have last lecture for today, last but not the least. I have a pleasure to introduce Professor Alan Walker. I gave the full introduction this morning, and he's a professor of pediatrics and nutrition at Harvard Medical School. And he will be talking on breast milk and microbiota in protection from necrotizing enterocolitis in prematures. Please, Alan.
Thank you. It's nice of you to persist, and it's always difficult to give the last lecture, so I'll try to go through it very quickly. I want to talk to you about breast milk and microbiota in the premature gut as a potential mechanism for preventing necrotizing enterocolitis. First, before we consider this, we have to consider the clinical problem, and then ways that have been used previously to prevent NEC, such as the expression of mother's breast milk and probiotics, and then a different approach, which is probiotics in breast milk-fed infants, before we summarize and conclude. Let's begin with the clinical problem. We all know how devastating necrotizing enterocolitis is. Even if the infant survives, it has lifelong problems. So it's very important that we prevent rather than treat the condition. 10% of babies under 1,500 grams develop necrotizing enterocolitis. It represents 1 to 5% of NICU admissions. In greater than 90% of cases, it occurs in premature infants, and it occurs very commonly 7 to 14 days after delivery at a time when the infant has been fed. And susceptibility is inversely related to gestational age. Now, this looks very familiar because it was taken from the same article, and Joe and I wrote a review article on that. This is a patient with necrotizing enterocolitis and pneumatosis intestinalis. 30% of patients with NEC require surgery. Surgery invariably leads to short bowel syndrome, and about up to 50% of babies, whether they're operated on or not, die of this condition. Sequelae are enormous and are lifelong. Short stature is very common. There's delayed neurologic development, particularly in cognitive function. All of these have a lifelong persistent problem, and the medical costs are prohibitive, costing about $2 billion per year. Several years ago, we hypothesized that necrotizing enterocolitis was caused by an immature, inappropriate, innate immune response to colonizing bacteria in the gastrointestinal tract. To support this hypothesis, we used organ culture of intestinal biopsies from newborns and then tissue obtained from therapeutically aborted fetuses, and we challenged these tissues with lipopolysaccharide and IL-1 beta, which are common causes of necrotizing enterocolitis. As you can see, the IL-8 protein level and the gene level was much higher in fetuses than it was in the neonate, suggesting that the excessive immaturity of the intestine was a factor in the hyperinflammation. Now, the innate immune response, inflammatory response, occurs when bacteria interact with toll receptors, which trigger signal transduction molecules, such as MYD88, TRAFT, and TRAC, and it leads to a transcription factor which enters the nucleus and causes transcription of inflammatory cytokines such as IL-8, TNF-alpha, and gamma interferon. Now, under normal conditions, this is a self-limited process because there are other components that interfere with the signal transduction and inflammation that address different aspects of the signaling process. We decided we would look at the components of the innate immune response in mature intestine, in intestine from aborted fetuses, and intestine from the resected portion of necrotizing enterocolitis. And as expected, we saw a strikingly elevated IL-8 response in immature intestine and in the necrotizing enterocolitis patient. When we looked at the signaling molecules and the receptors, we found that in each instance, TLR2, TLR4, MYD88, TRAFT, and NF-kappa B were elevated. What was most interesting was that when we looked at the negative regulators of the innate immune response causing a control of this response, we found that mature intestine had higher levels and immature intestine and necrotizing enterocolitis considerably lower levels, which we felt contributed to the hyperinflammation that occurs in necrotizing enterocolitis. All right, with that brief introduction, let's consider approaches to the prevention of this disease. We know, and Joe has already talked a little bit about that both breast milk and probiotics have been used to treat the condition. This is that meta-analysis that Joe referred to, showing that in small numbers of cases using different probiotics at different doses, that there appears to be an improvement. But you heard the other aspects of that he discussed, so I won't raise that right now. There's also a meta-analysis of breast milk studies. This is mothers express breast milk fed to the infant. And as you can see, in each instance, this was very successful. Now, we know and appreciate that breast milk influences the development of mucosal immunity. It does so through the microbiome, which we heard about earlier today. It does so through protective nutrients such as prebiotics and anti-inflammatory factors such as lactoferrin and omega-3 fatty acids, which we'll hear about tomorrow, and immune-enhancing substances such as nucleotide. What you might not appreciate is that breast milk acts as substrate for colonizing bacteria to produce metabolites that are anti-inflammatory. Now, we did a study looking at the influence of breast milk, infant formula, and then the combination of banked breast milk plus infant formula. As you can see, this is different in each of these circumstances. We also did whole genome sequencing, and we recognized that the genes from breast milk in the microbiota were different from those of formula. So we decided to look at what happens when probiotics or colonizing bacteria interact with breast milk. The reason we did this was we knew that tryptophan-rich foods that are fed to animals or adults can produce metabolites which are anti-inflammatory in the intestine of experimental colitis patients as well as the actual IBD patient. We also know that when high fiber and complex carbohydrates are fed to individuals or into animals, that these, under the conditions of bacterial colonization and certain bacteria such as Bacteroides fragilis can produce short-chain fatty acids, which by a variety of mechanisms are important in gut homeostasis. For example, fiber fed to animals produced short-chain fatty acids. These short-chain fatty acids interacted with G-coupled protein receptor, in this case 43, that are present on T regulatory cells, causing an increase in the expression of these cells. This is the microbiota itself, and these are specific and mixture of short-chain fatty acids, all of which cause an increase in T regulatory cells, as opposed to germ-free that has no increase at all. It also caused an increase in IL-10, which is an inflammatory cytokine. Now, we were most enthused about this approach to prevention when we noted in a paper that appeared in Pediatric Research that when probiotics were used with breastfed infants, they were much more effective than used with formula-fed, and depending on the degree of breastfeeding, it increased the dependency on preventing necrotizing enterocolitis. We had previously shown that Bifidobacteria infidus secretions free from the bacteria were anti-inflammatory. And what we did was we separated bacteria from the media itself. We then used fetal intestinal cells, which we pre-incubated with various fractions of the media, and we showed an anti-inflammatory effect that occurred. Now, when we used the less than 3K and 3K fractions in collaboration with the group in Salerno at the Research Institute in Salerno, we found that the 3K and 3 to 10K had a peak that didn't exist in the media. Now, using a database, we found that this was indolactic acid, which is a metabolite of tryptophan. We know that breast milk has large quantities of tryptophan, and so we felt that this was a potential source of anti-inflammation in the patients with necrotizing enterocolitis. What we did was we repeated the studies using fetal cell line, incubated with various concentrations of indolactic acid before we challenged with IL-1 beta. And as you can see, there is a striking reduction in the inflammation. To confirm our in vitro observation in human fetal cells, we used intact mice, both fetal and mature, and found that we could repeat this approach using fetal mice, but it was not the same as mature mice, suggesting that it was a specific fetal effect. Now, we know that indoles from tryptophan interact with aerohydrogen receptors, which can function as a transcription factor to produce IL-8. But when we used an inhibitor of these hydrocarbon receptor, we found that we lost the anti-inflammatory effect, strongly suggesting that this was the mechanism by which this occurred. Now, let me try to summarize what we have shown here. we have shown that when breast milk, which contains a large amount of tryptophan, interacts with Bifidobacteria infidus, this produces indole lactic acid, which is then transported across the fetal enterocyte, interacting with the aryl hydrogen receptor and causing a down-regulation in inflammation. We also know that complex carbohydrates in breast milk interacting with certain bacteria such as Bacteroides fragilis can produce short-chain fatty acids. What we didn't know was, do short-chain fatty acids have an anti-inflammatory effect on fetal enterocytes? So what we did is we incubated different concentrations of acetate, propionate, and butyrate, and found that each caused a reduction in inflammation, suggesting, again, an anti-inflammatory effect. When we looked at the mouse model, we found that the anti-inflammatory effect persisted in the small intestine as well as in the colon. And looking at the mechanism, we know that short-chain fatty acids affect deacetylases, causing a reduction and therefore reducing their epigenetic effect on the inflammatory cytokines. So here we show that when we used the 3 short-chain fatty acids with 2 fetal deacetylases, that in both instances they reduced the effect. We also know that short-chain fatty acids work through G protein coupled receptors. And in the case of the neonate, this is a 109A G protein receptor. And what we showed is that when these short-chain fatty acids were incubated with fetal cells, that they caused an increase in this receptor, and that when we used an inhibitor of the receptor, it suggested that we lost the inflammatory effect, suggesting that 109A was the receptor for this process. Now again, let me try to review with you what we've shown. We've shown that IL-1 beta, which is a common stimulus in necrotizing enterocolitis, can pass through its receptor and cause an increase in deacetylases, which by an epigenetic effect causes an increase in IL-8 and increased inflammation. Short-chain fatty acids through the 109A receptor can cause a reduction in the deacetylases, less of an epigenetic effect, and therefore affect inflammation. So these are 2 examples of how bacteria acting with breast milk in a different capacity can produce anti-inflammatory effects. So let me try to summarize and conclude what I've said. I've said that necrotizing enterocolitis is a devastating disease that must be prevented and not treated, that the best way to prevent this is to use breast milk in conjunction with a probiotic, and that although breast milk prevents NEC by a variety of mechanisms, including oligosaccharides and positive substrates, it also works by producing anti-inflammatory probiotics, metabolites. And then I've suggested that breast milk tryptophan metabolizes B infidus, metabolized by D infidus, produces indole 3 lactic acid, which is anti-inflammatory, but only in the fetal intestine. And also that breast milk interacting with other substances can produce metabolites like short-chain fatty acids, which are anti-inflammatory. Thank you very much. Questions? Yes, Jerry.
Of different compounds in the milk with anti-inflammatory activities and so on. How about IgA? Do you remember Martha Abel in Vienna?
Yep. No, but I'm now interested at this point in how probiotics metabolize breast milk. IgA may be a very important component, but I'm just looking at that side. Any other questions? Ray.
What is
Okay. Okay.
Do you see necrotizing enterocolitis in breastfed babies at all?
Yes.
And at what level do you see them?
Yes. Now, let me preface by saying I'm not a neonatologist.
Me neither.
Joe New is, but I'm trying to work out ways that we can do some preclinical studies that can then be done in large clinical studies that hopefully will help prevent NEC.
And the second question I had was, are there any animal models in primates where under natural circumstances you see NEC?
Joe? I don't know of any.
No, but I think the closest one is if you take, for example, a piglet, and if you do not give that piglet IgG, and you artificially feed that piglet, where you do not allow any mother's milk to that piglet, within 1 week that piglet will develop a bloated intestine, and it looks like necrotizing enterocolitis.
But, Joe, piglets are really different from humans.
Right. Yeah.
They don't have any immune function,
pigs,
and they require breastfeeding to get function. So I don't think that's a reasonable model.
I think that's the question you asked.
Yeah.
And the third question I had again is related to the underlying pathophysiology of what we call NEC.
Yeah.
What is the role of hypoxia in this situation? Because we are looking at a tube, which creates a lot of holes, and you put a lot of air out of it. I thought there were some early observations which suggested hypoxia may have a very important role in the induction of.
Yeah, I agree with Joe that this is a very complex disease, and that there are lots of factors that are involved. Hypoxia has been known. I'm just looking at one aspect of it.
But couldn't you explain a lot of these things which you see purely because of cellular damage? All the IL-8 and other lymphokine secretions may be purely related to the cellular damage rather than changes in microbacteria.
You have excessive IL-8 in necrotizing enterocolitis and immature intestine. So I think those are factors that are involved. Right. But I don't mean to imply that the only situation is an immature innate immune response. But it is true that in human prematures and in necrotizing enterocolitis tissue, that this is the case in terms of an immature innate immune response.
Are there some characteristic features of microbiota composition and function, it means metabolites, described in infants with necrotizing colitis?
There is. A number of people, including Joe New, have shown that there is a change in the microbiota before necrotizing enterocolitis occurs. And he explained that in his talk, that there are changes in the microbiota prior to the institution of necrotizing enterocolitis. Isn't that true, Joe?
Is it not connected with some real characteristics? It means that there is existence of some species which are not in healthy premature children.
Well, that's okay.
Go ahead.
Yeah. That does seem to be the case, that you have a dysbiosis with a shift in the two different phyla. One is an increase in proteobacteria, which is largely the gram-negatives, and the other one is a decrease in the firmicutes, which is gram-positives, those bacteria that are butyric acid producing some of the same metabolites that Alan was talking about.
Now, we have previously reported that the commensal bacteria or bacteria that are good bacteria can cause inflammation in the fetal intestine. So I think that's part of the problem, that response is abnormal.
One thing, in case of preterm low birth weight, whether any early feeding can give rise to the necrotizing enterocolitis, any feeding problem that related to the necrotizing enterocolitis?
Yeah, I'm not a clinician. So what I'm doing are preclinical studies. And before we can recommend what we do with preemies, we have to do a clinical trial. And that's something that has to be done. I'm just pointing out preliminary observations that suggest that breast milk in combination with a probiotic may be effective in creating an anti-inflammatory effect.
Okay.
Yes.
Yeah. Do we consider the difference between the real breastfeeding and breast milk composition? Because I think, okay, maybe the real breastfeeding is also different from the breast milk because there is some skin contact, maybe benefits for the premature immune system.
Go ahead, Joe. Do you want to talk?
So, yeah, I did bring that up a little bit in the talk that I gave, where the skin-to-skin contact and the enteromammary system that was described so nicely in the late 1970s by Alan and colleagues. I think that does play a role, and it's. But as far as the evidence is concerned behind that, I think that we still don't have hard evidence. We have good associations. We have the studies that were done in Israel, also in Western Australia, by Hasatu and colleagues, where it appears that this enteromammary system actually does occur, which necessitates close contact between the mother and the baby. And this is one of the reasons why in neonatal intensive care units in many parts of the world, we're trying to encourage the mothers to come in and to snuggle with the babies, have the babies on their breasts, and not just feed them the milk itself, but to have that direct skin-to-skin contact.
I should point out that mother's expressed breast milk is far superior to banked milk because it has so many different factors that aren't gotten rid of when you pasteurize. And actually, mother's breast milk from breast banks are later breast milk, and it doesn't contain a lot of the very important protective factors that exist with colostrum and mothers who deliver prematurely. Any other questions? Okay, we have ended early because we are short one of our speakers. And Natalie, do you have some comments to make?
I always have something to say, and first of all, I want to thank everyone being in this room. And I would like to thank people who are watching this session. I don't know how many, but I can tell you that yesterday's session has been watched by 1,334 people. So impressive. We can open a competition, huh? Which session will be watched the most. Okay, so we are finishing now, and we have a little bit time between now and dinner. For dinner, we meet at 7 o'clock at the entrance of the hotel. It's 5 minutes walk to the place where we will have dinner. And for you, we will organize again the transfer at 8:15 probably. So you have time. Weather looks to be nice. We are very close to the center of Lausanne. And if you would like to spend this time over there, it's just next door. There is a metro. You can take 3 stops to go to the place which calls Flon. And there you can spend your time and money. Yeah. And then if you're back by 7 o'clock, then the shops are open till 6:30, 6:45. So and by 7 o'clock, we're going to dinner. See you later.
Thank you, everybody who spoke.